changing world. And in higher education, another thing we are getting about the innovation, innovative thinking. So we have to think. Ideas are coming very fast. So we have to incubate it so that we get some start of the mentors. So always proper mentorship should be there in the teaching. We all the teachers we have to support the. And nowadays, another thing we have to manage is what I have observed, that stress. It is coming. Always stress is coming, particularly. We are looking at traditional medicine, particularly Ayurveda, which is very popular in India. I'm not talking about the treatment generation. I will say a few words about the Ayurvedic drugs scenario. But regarding Ayurveda, we are getting some good practices like balanced diet, good nutrition, proper sleep. Also, we are getting the yoga, meditation, and overall healthy lifestyle. So, if all these things will follow, then we could manage the stress and we can move smoothly in the higher education. So, since there at time before discussing on my topic, so few things I told about whatever idea I'm having, I shared it with the participant. And regarding the previous topic, the topic is scientific evaluation of ethnomedicinal plants perspective for effective therapeutic agent. So, if we look at the present scenario in the modern medicine and whatever drugs we are getting in the pharmacopoeia, particularly we are getting in the Indian pharmacopoeia, mostly we are getting the chemical drugs, synthetic drugs. We are getting also many biological products. But now we are getting many herbal products and phytopharmaceutical products. And if we look at the discovery or invention of the other drugs like synthetic drugs, even the biological products. In India, particularly, the contribution is very less. Even many people, they are doing very good work. So, B.B. Subodhi. So, I feel proud for him. He is an alumnus of Berhampur University. He is doing very good work in the synthetic drugs. But as far as the discovery of synthetic drugs or biological products in India is concerned, so we are having very limited contribution. We are getting most of the contribution from the Israel. Nowadays, a lot of is going on in the Israel. But in other developed countries like USA, European countries, we are getting a lot of contribution. But as far as the herbal drug is concerned, so India is very strong because we are having a tradition for the usage of the herbal, particularly the ancient literature. We are getting so many things about the use of many natural products, particularly the plant. We are having a traditional system of medicine, particularly it is also officially recognized. We are getting the Ayush. So it is officially recognized. And in Ayush system, many plants, many natural products, especially the plants, are used as drugs. So nowadays, whenever it will be utilized in the modern medicine, we have to apply the proper scientific tool. Then only it will be recognized in the modern medicine. But nowadays also we are doing a lot of research is going on in different laboratories in India, particularly on the herbal drug, and lot of methods we are developing for the standardization of the herbal drug. So if we look at the scenario of the IP, I have observed that different year initially in the IP, there are many herbal drugs. Then there is the question of standardization. So because of the standardization, gradually the number of herbal drugs reduced in the IP. But now, last few years, because of the policy of the central government, again the number of herbal drugs is increasing. Again it is increasing, and at present what data I have collected, exactly 169 herbs and herbal products and 7 phytopharmaceutical preparations are recognized in the IP. So I was also discussing about this thing with Professor Pulam, who is also with many high committees in central government is there. Now the government has taken a decision that each plant or any natural product, it is having some traditional use, and we are doing the scientific validation. And accordingly also we are getting the benefit, and it has been proved scientifically. Then also some of the constituents has been recognized. Some of the constituents are identified. Then it will be recognized as a drug in the modern medicine, and it can be included in the IP. So in this way, it is very encouraging. Day by day, we are getting more number of herbal drugs in the IP. There is a lot of research is going on, but it is not yet very clear regarding the herbal drug. Like synthetic drug, we are getting a proper protocol. In herbal drug, it is not there. Standardized method also differs from country to country. Also, different laboratories there have been different views. But we are carrying out some work and we are getting some success. And this is also a very nice branch of science, because initially information we are getting from our ancient text, from our traditional use. Then we are applying the modern scientific technique. And if it is proved, then we are using it in the modern medicine. So we are getting the merging of technology. We are getting the merging of ancient technique with the modern technology. Now also in different way we are using the herbal drug. Like many drugs which are used in the modern medicine, they have also been derived from the plant. Directly or indirectly they are derived from the plant. And this entire group is known as the ethnopharmacology. On the basis of ethnic claim, we are selecting any plant or natural product. We are carrying out scientific research. If you are getting positive response, it is in parallel with the ethnic claim, then we are using it in modern medicine. And many drugs, like we know that we are using the digoxin, we are using the quinine, we are using the chloroquine, we are using the artemisinin. So all these are, even though at present we are not deriving it from the plant, because when we are isolating from the plant, it is a very tedious process. Cost will be very, very high. Then also we are getting many plant materials that are also becoming day by day very extinct. That's why we are preparing synthetic agent. But the origin is the plant. So this is one way. We are getting the pure compound and we are using it as a conventional medicine. Then nowadays, the real herbal drug, we are getting the phytopharmaceutical preparation. We are getting the extract, we are getting some fraction of extract, we are standardizing it properly. At least some of the constituents. It is not possible. When in the extract, we are getting so many constituents. It is not possible to find out all type of constituents or analyze all type of constituents. At least some of the key components we are analyzing. Then, so it is known as a standardized extract. Then we are converting it into suitable dosage. Now there is also, along with the conventional dosage form, we are giving emphasis on the novel drug delivery system. We are also developing many novel drug delivery system. Particularly, we are getting the name phytozo. So by using the phytoconstituent, we are developing some modern drug delivery system, which is known as phytozo. Also, we are using the word herbozone. So all these things, because of we are using the natural for development of this novel drug delivery system. So these things are going on. So during my presentation, since I am addressing the teacher, there are many common slides. So in detail, I will not discuss, but some of the important slides and some of I discussed about the background of this, particularly in this field. Particularly, I will discuss the common method which we are adopting for development of herbal drugs from the nature, and whatever detailed contribution, research contribution I am having in this particular field. Those things also I will share during my presentation. Okay. So this is very common. So we are getting different uses from the plant, and also we are getting some poisonous substances, toxic substances from the plant. So it is known to all the people. Next slide. So we are getting different type of uses like food, medicine, and spices due to the presence of primary and secondary metabolites in the plant. It is also very common. So whenever we are using the plants as drug, we are not. It is also very common that we are not using the whole plant. Sometimes we are using a particular part of the plant depending upon the accumulation of the active constituent in a particular plant. But that's why we are using the different type of plant. And some of the examples I have cited here. Next slide. So this is very important. Worldwide usage of the herbal drug, particularly traditional system medicine. So we are giving different names like alternative medicine, traditional medicine, and complementary medicine. So different part of the world, it is used in different ways. In African countries, they are having the traditional medicine, but it is not officially recognized. Also, we are not getting any standardization method in African countries, and we know that many poor countries also they are in the African countries. But in the countries like India and China, so we are having a strong traditional system medicine. It is officially recognized. In China, they are having the famous Chinese system medicine. Tibet, they are having the Amchi medicine. In India, we are having the Ayush system. It is also officially recognized, and we are giving emphasis on the standardization. And in developed countries like USA and European countries, they are also having strong herbal practice. Actually, they are not having any traditional system medicine. They are having a strong herbal practice, but they are not using the herb as drug. Mostly they are using as nutraceutical and health food. So this is the usage of the natural products and traditional medicine in different parts of the world. Next. So plant metabolism is very common that we are getting the primary metabolism, we are getting the secondary metabolism, and we know that primary metabolism particularly due to the requirement of the plant, they are dependent on carbohydrates, proteins, and lipids. Also, during the production of this primary metabolite, we are getting the production of many secondary metabolites, and we are getting a lot of uses. Always primary metabolites we are getting in higher amount, higher volume, and secondary metabolite we are getting in most of the secondary metabolites we are getting in low volume, but these are highly valued products. Next slide. So this is very common to the teacher that we are also teaching at the B.Pharm level, at the PG level, that how we are getting various basic metabolic pathways for the formation of all type of metabolites in the plant, and actually the plants have a fast chemistry. Actually, the human being, they learn chemistry only from the plant. So because of the autotrophic nature, because of the complex enzymatic system, they are preparing all type of metabolites. They are preparing all type of metabolites. So we are getting all type of basic metabolic pathways. Starts with the photosynthesis, we are getting the glycolysis, then we are getting the Krebs cycle, then also we are getting the cyclic acid pathway, we are getting the amino acid pathway, and from all type of amino acids, how we are getting the primary and secondary nitrogenous compounds. Then also we are getting the long chain compounds like fatty acids, and we are getting the lipids. Also another pathway is the mevalonate pathway. We are getting all type of corpuscle cell steroids. So in this way, we are getting all type of primary and secondary metabolites from the plant because of the autotrophic nature of the plant. Next slide. So chronologically, I have shown the isolation of different phytoconstituents. So morphine was the first bioactive molecule which was isolated in the year 1806. Then onwards, we got the isolation of many compounds, and particularly during the isolation of salicylate from the salic salva and the production of the salicylic acid in the year 1838, it was a very important time. So, particularly salicylic acid, it was converted. The distillation, it was converted into the acetyl salicylic acid, which is known as aspirin. Till then, aspirin we are using it as an analgesic drug. So, after that, scientists throughout the world, during that time, actually the herbal drugs were mainly used as the medicine. But after that, the scientists all over the world, they tried to find out the bioactive molecules which are present in the plant. They tried to isolate it, and also sometimes they did the structural modification, and they were used in the conventional medicine. Next slide. So again, these are the isolation of different constituents, and particularly we know that the work on actinium. So one scientist, Wu Yoyu of China, he got the Nobel Prize in the year 2015 because of the work on the actinium, and we know that it is very common. Even before that, natural product we are using against malaria like chloroquine. But gradually what happens, we got many chloroquine resistant malarial patients. So again we solved the drug, and that also solved drug we got it from the Chinese plant, that is Activisia annua. We got Activisia and we are using it in the form of Accredited and Accredited. So everything is there in the nature. Even whatever drug we are getting in the conventional medicine, directly or indirectly, they are also derived. Some connection is there with the plant. Next slide. So these are the common things. So herbal drug, we are getting many advantages. Main advantages we are looking at, we are getting less or negligible adverse effect. That is why everybody is going for the herbal therapy. And the world is again going back to the herbal therapy. Next slide. So acceptance of herbal drug is everywhere because of many qualities. Next slide. So whenever we are using the plants, natural product, particularly plants are dropped in three ways. Particularly we are using the plant. One is the homemade remedies. So according to our tradition, so whatever we are getting from our forefathers, accordingly we are using some plants. Just we are taking the plant, either directly we are taking it, or we are squeezing it, then we are taking it. So this is the common homemade remedies. Then also sometimes we are isolating a particular constituent, then we are standardizing it, then we are using it in the conventional medicine. But the real herbal drug is the phytopharmaceutical preparation. We are preparing the extract or we are taking the fraction of the extract in a standardized form. Standardization, actually this is the meaning for different people, they are in different meaning. What in standardization? At least we have to characterize some of the components, at least the components which are responsible for the biological activity. What happens in the plant whenever we are taking an extract? Sometimes we are getting additive effect, sometimes we are getting synergistic effect, and when we are taking a whole extract, what happens? Same drug we are getting the adverse effect. But in the form of extract we are taking, that adverse effect is nullified. Adverse effect is nullified. That is why we are preparing the extract. But standardization is a real problem. That is why the proper standardization it will be recognized. Next. So again, that's something I have discussed here. That whenever we are getting many components, particularly that are targeting different type of receptors, we are getting additive effect, we are getting synergistic effect. But whenever we are getting many constituents, standardization is a real problem. Because single drug, even in pharmaceutics, many of the faculties, many of the teachers, they are carrying out the work in the pharmaceutics. Release pattern when we are studying. In a single drug we are taking, we are developing any dosage form, then we are observing the release pattern. It is very, very easy. But in case of herbal drug, when we are preparing any type of dosage form, we are checking the release pattern. It is not that much easy. Because many constituents are there. Which constituents will target? So that is why it is very difficult. But when we are using the extract, we are using the herbal drug, since we are getting many advantages, that is why day by day the use of herbal drug is also increasing. Even the developed countries, they are also using it. What proper standardization, say presently in USA, US FDA is very strong. What some herbal drugs at least they are recognizing after proper standardization. Next. So whenever we are getting, suppose after extraction we are getting a single component it is having, because many students they are presenting very trace amount, not possible to isolate it. But if it is possible to isolate it and we are isolating it, three ways we are using. If the yield is very less, we are preparing it by chemical synthesis. Many drugs are doing like paclitaxel. It is well in the plant. But is it possible that we are just isolating it from the plant? Yield is very less. Vindication in plastic. It is present in the plants manufacturing years. Yield is very less. That is why we are not isolating it, we are preparing it by the chemical synthesis. So if yield is less, we are preparing it by the chemical synthesis. And sometimes we are also carrying out the structural modification. You know that many drugs we are doing the structural modification. So Taxol we are doing in the form of paclitaxel. We are doing in the form of artemether. Like that, porophyllotoxin we are using the etoposide, tenoside. So many examples are there. We are carrying out the structural modification so that the biological activity will be enhanced and the adverse effect will be minimized. Then also, whenever the yield is more, then also we are expecting it. Like some examples are also there in the India, like kurkumi, yield is good, we are expecting it. Berhampur, yield is good, we are expecting it. That's the name. Yield is good. We are expecting it. Then we are using it. So if the yield is good, then we are expecting it so that it will be commercially viable. Whenever any production we are doing, it should be commercially viable. So that is why if the yield is more, always we are expecting it from the plant. Next. So here I have shown some slides. Particularly when we are carrying out the work in the field of ethnopharmacology. So it is a very good area. And sometimes on the basis of ethnic claim, we are selecting the plant, we are carrying out scientific validation, and in parallel with the ethnic claim, we are getting the activity. But in some cases, we are not getting. Some cases, we are getting some ethnic claim. Accordingly, we are carrying out the activity study, but we are getting some other activity. We are not getting the same activity. And sometimes, randomly, we are selecting the plant, we are carrying out many activity studies, and we are getting some activity. So, three ways, particularly whenever we are carrying out the work in the field of ethnopharmacology, sometimes in parallel we are getting the activity, it is good, so that ethnic claim is there, accordingly we are getting the activity. Sometimes we are not getting, but we are getting some other activity. And sometimes also we are carrying out the randomized screening method, the famous drug, Taxol. It was also developed by randomized screening method. So here are some examples I have shown that Androphis paniculata, very common Indian plant. So it is having some calcium there, accordingly we are getting that. Same thing also Artemisia calcium it was given in the China against malaria. Similarly also getting various also same thing. Next slide. Here also other example is Cassia Pipilia, then Sycona. Sycona also traditionally it was given plant. We also got the similar activity. So Salix alba. So all these are the examples. We are getting the activity in parallel with the these are the next. So here again, we are getting some examples. So on the basis of ethnic claim, we are carrying out the activity. But according to the ethnic claim, we are not getting the activity. We are getting some other activity. Some examples I have cited here, and all the teachers know about this Cathanthus roseus. Cathanthus roseus, particularly in the Asian countries, African countries, it was a traditionally very popular drug against diabetes. And the scientists carried out research, but they didn't get a significant anti-diabetic activity. So even they lost hope on this plant that it is not having only we are getting some traditional case, but some scientists in the early league, they particularly carried out again they just extended the one. They got some very interesting indole derivatives like zincistin and vinblastine, and they scored very good activity against cancer. Now also it is a drug of choice. Even the yield is very less. Yield is very less. It is not possible to isolate it from the plant. We are preparing it synthetically, but the origin is the plant. So accidentally it was found. So in this way, sometimes even though we are not getting the activity in parallel with the extinction, we are getting some other activities and we are using it in the modern medicine. Next slide. So here particularly some examples are cited random because nowadays we are getting screening method, we are getting many constituents. At a time we are getting out many activities and we are getting different activities. Accordingly, we are getting some significant activity and activity they are being used. And even the famous we know that from the Texas very good, we are getting the Taxol by this randomized screening method. It was also developed. Next slide. So some structural modifications as already I have told that activating of instruments, again the research, always the research is going on in different laboratories and we are getting some structure. Suppose we are getting some demerits like regarding the activity, regarding the solubility, also regarding the adverse effect. So we are getting out some structural modification. In my research also I have done, we have isolated some constituents, we have carried out structural modification, and after structural modification, if you are getting the benefit, then that derivative only we are using. So many examples are there. It is also very common to the teachers of pharmacology. Next slide. So here are some example of the modern drugs, how we are getting it from the plant like Accredited. Interesting, we are getting such a complex structure. So we are getting the indole and indole nucleus, very complex structure, but effortlessly prepared inside the plant. So we should go before the nature. That how effortlessly. Scientists tried a lot of year only it was developed and nearly this 2 days they are maybe knowing that 22 to 23 steps is required in order to prepare this compound. But now we are preparing. Now we are preparing it because it is not possible since it is very less. Similar also case of deoxy, very beautifully designed molecule. We are getting the steroid. 700 position we are getting the lactulin. 3 position we are getting 3 digitoxin molecule. Then only we are getting the cardiac. Otherwise we will not get. But in the entire plant, again, after digitoxin we are getting another glucose. When we are drying it, this glucose will be eliminated. We are getting the secondary glycosidic form, which is only active as a cardiac. So very beautifully designed inside the plant. Next slide. So here are also some example of other patent. Next slide. Again, here we are getting the patent text. Very complex molecule. So actually, it is how it is prepared inside the plant. It is dietary in nature. Very complex molecule. But again, yield is very less. So we are getting alternative methods for preparation of the textile, particularly patent textile. So from the plant or from the leaf also, we are getting some other nutrients because it is present in the bark. Taxol is present in the bark. If you remove the bark from a plant, it's a tree. Taxol is a tree. It is the plant is available in the higher tissue. If you remove the bark from the plant, then the plant will die. So we have to destroy a lot of plant material. That is why we have developed some alternative ways how to prepare the patent textile. Then next. So, as already I have told, the Chinese scientists got the Nobel Prize because of our work on the activity. So, now in the clinical medicine, so we are using the use of method. So, this is a usual approach. How generally, very simpler way in a single slide I have shown it, but it is not that much simple. It is very complex in nature. How we are selecting the plant. So, initially we are selecting the plant. For selection of the plant also, it is not possible. It is not the work of a single discipline. Actually, always this type of work, it is a multidisciplinary approach. So we have to take the help of the experts from other disciplines. Selection of the plant, we are not having the expertise. Sometimes we have to take the help of botanists, especially ethnobotany, taxonomists, even anthropologists. They can help for the selection of the plant. In my research, many plants are selected by collaborating with the taxonomist and with the ethnobotanist. So selection is very important. Very judiciously, we have to select the plant, and very correctly also we have to identify that plant. Because regarding the identification also, that is where always we are putting the authentication certificate. Properly it should be identified. If the plant is not properly identified, then the whole work it is not having any value. So it should be properly identified. After that, we are carrying out the extraction. So after within the extraction, generally we are carrying out the bioassay. If it is having significant activity, then we are carrying out the thorough phytochemical investigation. So regarding the phytochemical investigation, it depends upon the facilities available in a particular lab. So in the laboratory grade, generally we are carrying out the chemical test, we are carrying out the PLC, we are carrying out the column chromatography, and nowadays, since we are getting many modern tools, particularly we are giving emphasis on the HCMS analysis, SCMS analysis, even LCMS analysis, and we are developing the HPLC fingerprint. Because we are not using HPLC and GC, it is not very suitable for the herbal drug. We are using the HPLC. Nowadays we are getting a lot of modification in the HPLC. So HPLC also we are using, and after that, if we are properly standardizing it, then also we are getting good activity study. Then we are getting the standardized extract. And standardized extract only we are developing into suitable dosage form, maybe conventional dosage form, maybe low delivery system. And after that we are going to the clinical trial. But reaching up to this stage, not that much easy. Many failures will be there because whenever we are carrying out any type of research work, very honestly. Suppose I am carrying out any type of plant I am taking and carrying out some activity study. If the activity is not coming, that also I should report it. But I should not do any type of manipulation. Otherwise, it will create a false impression. That is why nowadays also we are getting, jokingly I am telling, we are getting one journal, that is Journal of Negative Result. Maybe because of this reason also this we are getting this journal of negative result. Even though we are getting the negative result, it is also a research work. We are carrying out the study, we are not getting it. So very honest. So it is my request to all my young friends, whenever we are carrying out any activity study, very honestly report it. Even if you are not getting the activity, you just report it. So this type of study, very simply I have shown it, we are getting many failures. And if we are getting success, then we are going up to the level of clinical trial. So that it will be used in the modern medicine. Next slide. So selection of plant, different approaches have there. Like total random is sometimes selective. Then chemotherapy is significant. If one genus is showing some activity, other species coming under the same genus, they may have the similar. On that basis also, in my research also, some plants have selected on this year. Then geographical difference plants are available in different part of the world. If one plant available in one region it is showing some activity, same plant available in other region also we are testing it. Then good thing is the traditional medicine. On the use of traditional medicine, traditional use and ethnic level and tribal, because many tribal people now also they are depending on the basis of tribal claim, ethnic claim, folkloric claim. Generally, we are selecting the plant, and many plants are selected in my research on the basis of this ethnic claim and tribal claim. Next slide. So, usual approach, some deviation is also there. In which season we should particularly collect which part of the plant. Next slide. So these are the extraction techniques. Generally in general laboratory, we are getting the academic institutions, we are getting the ventilation, population, substrate extraction, and the practical principle we are doing the distillation. But nowadays we are getting some advanced this extraction techniques like microwave assisted techniques in some laboratories. They are keeping this microwave assisted extraction technique. Less time is required, less volume of solvent is required. Then also very specific extraction. Suppose I am carrying out any extraction with alcohol, we are getting so many constituents. And whenever I am getting a mixture, very complex mixture. But specific extraction purpose, we are using a microwave and supercritical food extraction. Lot of applications we are getting in the food industry. Also for the volatile extraction, we are using it. Nowadays, for preparation of the delicate perfume, we are preparing the toxin less volatile. For that purpose also, we are using the supercritical food extraction, and you guys know this technique very well. Instead of using the classical organic solvents, we are using the supercritical carbon dioxide. So carbon dioxide will act as a free flowing liquid. Next slide. So then this is general things. So getting the dietary extract. So we are considering it, we are getting the preliminary screening, and generally we are getting the chromatographic analysis. Next slide. So these are some of the techniques that are shown, very common. Many laboratories having this facility. Next slide. So bioactive activity by vaccination and isolation of the active compounds. So whenever the extract is showing any activity, we don't know what is the mixture. So many constituents are present. So we have to find out which constituent is responsible for the biological activity. For that purpose, we are carrying out the bioactive vaccination method, which is a very, very tedious process. When the whole extract is showing the activity, then we are fractionating it. Each fraction, again, we are carrying out the activity. The fraction which is showing good activity, from that fraction, we are trying to isolate the constituent. And if we are successful, then only we are getting the activity. But sometimes also we are not getting success because large amount of the plant material is required. When we are getting large amount of the plant material, we are getting some extract. Again, we are fractionating it, and when we are isolating it, we are getting very few, very trace amount of the compound. So it is a very, very tedious process. Generally, we are carrying out it by the solvent extraction method or by column chromatography. Again, column chromatography also lot of skill is required, and also it is a tedious process, very time-taking process. Nowadays, particularly in I research previously I was doing column chromatography separation and I was isolating the constituent. But nowadays, since many modern techniques are available, like GCMS, particularly LCMS. LCMS is very advantageous LCMS. So in order to fractionate the extract and in order to get about the different type of constituents which are present in the extract. But this is the usual process by which from an extract, when it is showing some activity, which constituent is responsible for the biological activity, that also we are finding out. Next slide. And sometimes also we are facing difficulty that many times we are getting a synergistic effect. If many constituents are present, then only we are getting that. So that case, it is not possible to isolate a single constituent. So in plant, that complexity is there. Sometimes single constituent is responsible for the biological activity. sometimes a group of constituents, they are responsible for the violation. So whenever we are getting the result, accordingly we should interpret it. Next slide. Next slide. So because of the biotic extension, we are getting some compounds, biotic compounds, and particularly lead handling process also we are using it, and sometimes also we are using it is the marker compound for the standardization of the. So quickly I'll try to. Something might be just tell me. Next. So schematically, I've shown it how we are taking the whole extract, we are fractionating it, fraction we are getting out the activity, we are getting the isolated constituent. Then after getting the isolated constituent and it is showing the activity, how we are getting out the chemical modification and protein. Next slide. So generally, these are the common that we are getting the field in the study like in silico. Nowadays, we are getting the in silico study because of the computational biology. In vitro, in vivo, study and clinical trial at the end we are doing. Next slide. So very common in vitro and in vivo techniques as to it is very common. Next slide. Next slide. So these are some of the plants. These are some of the plants I have selected in my research and we have prepared the study. Particularly some plants. Significant work I can have just I'll tell it that we are getting the Acredited. So this plant was selected for a biotechnology scholar. So one plant biotechnology. He was also one of the supervisors. So we took the plant. With the intact plant, we carried out the extraction and we did the thorough phytochemical study and also we carried out the anti-diabetic activity. Then we developed the in vitro propagation, particularly hairy root culture. So that facility was available that time in the Utkal University. So we developed the hairy root culture. So that hairy root culture, that in vitro propagation, we also took it and also similar type of comparison we did. We did the phytochemical work, we did the inorganic matter analysis, we did that anti-diabetic activity study, and we compared it. So this type of study was carried out with the SIDA. Again, other plants also we experienced for the anti-diabetic activity regarding the stabilized asphalt. Stabilized asphalt from the bud, we isolated alpha-amylase acetate. So after its isolation, it also showed very good anti-diabetic activity. So we got it in good amount and it also showed very good anti-diabetic activities. Again, we took it as a master compound. And its content was estimated in different plant parts, particularly in the bark, in the wood, and in the leaf. So after experimental analysis, we got that maximum amount is present in the bark, less amount is present in the wood, and also it is not present at all present in the leaf. So this type of study also we carried out the stabilized asphalt, which is common in the next slide. So here on the basis of chemotaxonomic significance. So one plant is selected, particularly Chlorodenum philippinum, in the Jashpur district, in the Keonjhar district, in the Baldwin district. So traditionally it is used against diabetes. On that basis, we selected the plant for anti-diabetic. But some other genus of the other species of the same genus, Chlorodenum, we also selected and we carried out the anti-diabetic. But among all those plants, Chlorodenum philippinum, it scored maximum. Next slide. These are some of the weeds also. We have taken some weeds. We carried out anti-diabetic activity. Next slide. So here particularly anti-cancer activity study, we did all these plants we explained, particularly against the lung cancer and breast cancer cell lines. And among all these plants, Lantana camera is for the most significant anti-cancer activity. Next slide. So this is the usual approach for the standardization of the herbal drug. Next slide. So nowadays we are giving emphasis. One of the standardization parameters we are giving emphasis on the development of chromatographic fingerprint and also quantitative estimation of the marker compound. So these we are taking as a standardization parameter to standardization herbal drug. In my research also, I did some quantitative estimation of the marker compound as well as I have developed some chromatographic fingerprint. Particularly, I have chosen the HPGA technique. Next slide. So how we are taking a marker compound, sometimes we are taking a common marker, chemical marker, sometimes we are taking the biomarker, and sometimes we are taking the bioactive marker. Very difficult to get the bioactive. So we are taking the common marker or we are taking the biomarker. Next slide. So these are the example of some of the common markers in common medicinal plants which are used in India. Next slide. So generally three techniques we are following: HPLC, GC, and HPTLC. But most suitable for this purpose is the HPTLC because HPLC and GC lot of restrictions are there. So HPTLC mostly we are using it since it is a Panaga model technique. Next slide. Next slide. So this is the HPTLC. When I use HPTLC in my research, that HPTLC was different. But nowadays, lot of modification is there in the HPTLC. Last time I had been to ILS, they purchased a HPTLC, and I was in the technical committee. So I got that. But isolation method also nowadays it is possible with the HPTLC. From the plate, if you are getting some significant constituents, so directly it will be taken by a tube, and the compound will be isolated. Directly also we are getting its mass spectra and NMR spectra, and we are getting the characteristics of the compound. So this is also very accurately possible with the modern HPTLC. So I got it from the ILS. Next slide. So some examples. Next slide. So particularly I have carried out the quantitative estimation. I have taken some marker compound. So whenever we are doing any type of analytical technique, so we are carrying out the validation of the method. So all these things I carried out. Next slide. Next slide. So in a chromatogram, we are taking the previous slide. Chromatogram, we are taking the standard compound, then we are taking the samples. So how we are taking the marker compound, then in the standard also we are taking the marker compound. Next slide. So this is the HPLC chromatogram. Next slide. Next slide. Next slide. Calibration because always for the quantitative estimation we are developing this calibration curve. Next slide. So how we are getting the presence of this 6 ginger, how it is present in different samples, so it has been stored. So in this way, we are carrying out some other examples of next slide. Next slide. Next slide. Next slide. So similar technique. Next slide. Next slide. Next. So here particularly alpha amyloid acetate. So how it is present in different plants. So we have estimated. So by carrying out the estimated study also we are carrying out this. Also we are getting good publications out of this one. Next slide. Here I can see that about study. Next slide. Next slide. Next slide. Next slide. Next slide. Next. So here we are taking the berberine. The alpha berberine we are taking as the marker compound. And its contents was estimated in different formulas. Next slide. So how we are getting it is looking very brilliant, but we are getting very important information. In this way, we are developing the chromatographic fingerprint and we are getting important information. As I have told, in extract, not possible to separate all the constituents, not possible to analyze all the constituents, at least some of the components. And in a particular system, in a particular environment, if we are developing the chromatographic fingerprint, then also it is an important standardization parameter. Next slide. So nowadays, in silico study is very common. Many of the teachers, I think, also they are doing this in silico study. So we are getting the structure of many phytochemistries. So we are getting the ligands. We are getting the proteins, the target, and we are getting the docking study. And by this also we are developing the phytochemical database, and we are getting the information before carrying out the wet laboratory study. We are getting information about the many phytochemistries, how they are active, and also we are developing the mechanism of action. Like this method. So one of my scholars also is working particularly in the rheumatoid arthritis. So some plants on the basis of this docking study, we have taken some plants and is also extracting it. Some constituents are present, particularly indole derivatives and benzopyrene derivatives. These are responsible. So we have targeted these compounds, and also one of my scholars present also is carrying out the study. Next slide. Next slide. So, in conclusion, I can say that this is not a work of a single laboratory, single discipline. Always we should have an integrative approach. Always we should have a multidisciplinary approach. That is why in my research also I got the opportunity to supervise the research scholars in different subjects like Ayurveda, chemistry, biotechnology, botany, and microbiology. Also, I have carried out some research projects in collaboration with other subjects. At present, also I am involved in a research work in College University, one joint project, multidisciplinary approach. Even the subjects, social science subject like anthropology, it is also related. So one work particularly we have carried out with anthropology, here also from anthropology, it is also possible. Anthropology people, they are collecting information from the forest areas, like we are getting different type of traditional beverage. They got some traditional beverage, particularly from the Koraput district. So we got this traditional beverage and we carried out some type of scientific study. Like beverage means definitely ethyl alcohol is present. Percentage of ethyl alcohol, then this is GCMS analysis. We got the different type of constituents which are present. Then by putting it in the virtual lab, particularly ADMT lab, we got that which type of activity all these constituents are having. So this type of study is also possible because we don't have the expertise. So they are collecting information from the forest areas by interacting with the tribal people. They are getting the beverages, we are getting it, and whatever possible according to the facilities available. So we are carrying out some study, and also it is possible to carry out this scientific validation of this traditional use, traditional things. So that also we are carrying out. Yes, sir. So why I put this mantra here? Amantram aksharam nasti, nasti mulam nausadam, ayogya pulso nasti, yojaka sastra duplava. So this is very important for the picture. So whatever in the class, whatever we are speaking, it should have some value. As already we have developed, we have discussed any plan, definitely it is having some excellence. Any person, maybe any person, they are having some good health, good human being. We regarding this hard thing that any person they are having some good value. We are getting a very good story, particularly in the Mahabharata. That when both the Kaurava and Pandava, they were studying their Dronacharya. So after completion of their study, Dronacharya asked Duryodhana and Yudhishthira. He told Yudhishthira that you bring one bad person. He told Yudhishthira, Duryodhana, you bring one good person. Both returned empty-handed. First, Duryodhana told that I just interacted with many people. All are having some bad qualities. So I thought that I am not getting any good person. Yudhishthira told that I interacted with many people, but I got that they are having some good quality. So I didn't get a single bad person. So similarly, in a class we are getting many students. Even when I was a young teacher, we are getting many students. Sometimes also we are getting irritation because of some students, but all are doing good. So whatever students we are having in our class, all are having some work. So they are having some talent. They are also unique individuals. So in this way we should move, and particularly our role as a teacher is a yojaka, enough. So whatever we are speaking, how we are grooming the students. So accordingly we are getting the future of the country. So with all these words, once again I thank the organizers for inviting me as a resource person of this academic development program, and also I am really privileged to speak my ideas. Whatever little research work I have carried out during my career with my teacher colleagues. Thank you everybody. Thank you so much.
Good morning to all. First of all, it's my pleasure and privilege being the chairperson of this session of this scientific program. I would like to thank and express my sincere gratitude to our respected research person, Professor Sagar Mishra, coordinator, UGPS, Uttar University, for accepting our invitation and presenting so nicely in this occasion. The content is the presentation what Sagar highlighted is highly correlated and significant with respect to the topic concerned. So no doubt from ancient time until date, we are using the traditional plant for their therapeutic value for prevention and treatment of the diseases. But again, it has to be validated systematically and scientifically to be safe and effective. Because whatever the extract or the process of the using, it should have the therapeutic value within the limit. And Sir has nicely explained and highlighted about the identification and authentication of the plant, about the screening method, about the pharmacological, toxicological, and the clinical study, so thereafter it can be marketed for use. Thank you, sir, for sharing your experience, expertise, and giving your valuable time by traveling such a long distance from Bhubaneswar to Berhampur. So really, we are very much fortunate and thankful to you for providing such type of teaching methodology and the research work which can be helpful for our future. Thank you, sir. So now it's time for the presentation to so for that, sorry, sir. So now, so many things are as described, starting from the standardization of the different plants, the sources, advantages, limitations, about the chemical modifications, and how to identify all these things about the structural insulation. So I request if there is any query of the question from the audience, you may ask target details.
Thank you, sir. Namaskar. Thank you for a informative and interesting session. I have a simple query that in silico study reduces the time and help us quick completion of our work. But if we do not have any information about the structure, our the molecular structure of this thing, so is it possible to go by silico fast or fast we have to complete the structure?
Actually, in India, we are getting most of the plants have been phytochemically thoroughly reviewed, and we are getting many structures. Many structures we are getting. Even if we are using many plants in the modern medicine, but still there are many plant species which are unexplored. Previously, we are using chloroquine as a drug of choice in case of malaria. But as when we got the chloroquine resistant patient, again we develop a drug from the plant, that is IPGA. So similarly also the research work is going on. Then also many of the plant species have been thoroughly reviewed. Lot of phytochemical work has been carried out, particularly after development of the structural illustration method. Nowadays we are adopting the NMR technique, NMR, mass, X-ray crystallography, IR study. So many structures we are getting, many phytoconstituents we are getting the structure. So if we are not getting the structure, not at all possible. But most of the plant, even on the basis of suppose some plant has not been explored, we are getting a new plant. But similar species also we are getting some genus. We are also getting some other species, and that similarity is also there. If a species coming under a particular genus, it is doing some activity, or it is having some kind of constraints, the other species coming under the same genus, it is also it's having the same type of activity, same type of constraints as we are getting in case of solar. Solar family, particularly we are getting the propanol. Many plants coming under the solar family, they contain the propanol. Many plants coming under the family Rutaceae, they are also having the polar plate. Many plants, particularly coming under the umbelliferae, so the fruits they contain the polar plate. So on that basis, at least any plant at present, because so many work have been carried out, at least we are getting some plants, some information will get about the constituent. So when we are getting the structure of the ligand, so many proteins are available. I am not a very expert, but many people they are very expert on this in silico study. They are carrying out the study, and before carrying out the wet laboratory study, at least we are screening the plants. We are getting a statistical database. On that basis, we are selecting some plants. Another advantage is development of the mechanism of action. It is very easy. But because of the protein binding site, we are also developing achieving some type of mechanism. So this is also possible. So what anyhow structure is required. Without that, how it is possible? Structure is anyhow it is required. But most of the plants have been explored in India. Lot of research work is going on in different development.
Thank you, sir.
Anybody has any other?
Like when we go for this anti-arithmetic test, and basically the Google research or so the test that we carry out, particularly when doing the geometry fix, it becomes difficult on our part to carry out. So can you suggest any particular case which can be taken up for the anti-arithmetic standardization of the
Generation of what?
curriculum? Well, any curriculum.
Curcumin itself it is a isolated constituent. Curcumin itself is a isolated constituent.
So while taking this curcumin from different sources, it becomes difficult. Like suppose we are taking it from yellow turmeric and take it from that.
Maybe yield will be less. Again, curcuminoids particularly, it is a mixture of curcumin, dimethoxycurcumin, and bis dimethoxycurcumin. Three mixtures jointly we are calling it curcuminoids. If it is present in a turmeric species, so it will be present. Maybe yield is less. Like in my work also, I have studied particularly regarding the presence of 6 ginger in different species of gingiva personnel. So when we are collecting the plant from different regions, different yield of 6 ginger is there, but it is always 6 ginger. So similarly in your case, since we are getting amount of cumin, so it is a constituent. So how do we different? Yield is different. Yield is different in different species. And previously by isolation we are estimating it, but estimating it, some error may be there. Nowadays we are adopting the HPLC technique and other modern techniques are also. So am I clear what you are asking?
Thank you, sir, for a nice explanation. So now it's time for presentation for our respected research person. So for that, I would like to request to join Professor Mr. Sir, Professor Sudhir Sir, Dr. Bharat Sir, Professor Madhu Madam, and Professor Rajesh Madam. Please come to the dais and consider respected research person.
Thank you, Sagar sir, for the brilliant presentation. Now we are going to start the second science section, which will be delivered by Pedagogy teacher, respected Dr. Bharat Bhushan Subudija from Sikya Anusandhan University, Bhubaneswar. Yeah, I would like to invite our resource person, Dr. Bharat Bhushan Subudija, to please come on the dais and occupy the chair. I request for a total welcome and would like to invite our associate professor of this institution, Mr. Asit Baran Panikari, to introduce our resource person for this session. After this, we will continue this session with the class.
So good morning, one and all. It's an extraordinary privilege, a profound honor, and a moment of immense academic gratification for me to introduce a personality whose scholarly immense intellectual profundity and scientific acumen have illuminated the frontiers of pharmaceutical research. It is indeed a matter of immense personal pride, and I had the rare privilege of being his student during my M.Pharma program, where his erudition, pedagogical brilliance, and unwavering commitment to academic excellence profoundly influenced my professional journey. Our distinguished resource person, Professor Dr. Bharat Bhushan Subudhi, is an illustrious pharmaceutical scientist, an eminent medicinal chemist and accomplished academician, an innovative researcher, and an inspirational mentor of exceptional repute. He is currently serving as a professor at the School of Pharmaceutical Sciences, Shiksha Anusandhan University, Bhubaneswar. With an illustrious academic career spanning over 2 decades, Professor Subudhi has established himself as a paragon of scholarly excellence and scientific innovation. His pioneering investigations are centered on the rational design and development of novel therapeutic agents against emerging and multi-drug resistant infectious diseases through the seamlessly integration of computational drug design, medicinal chemistry, molecular modeling, and pharmacokinetic pharmacodynamic optimization. His research epitomizes the confluence of scientific ingenuity and translational relevance. Professor Subudhi has demonstrated extraordinary leadership by successfully conceptualizing and executing numerous high-impact research projects funded by India's most prestigious scientific agencies, including DRDO, SDRPTST, DBT, ICMR, and DIRS. These competitive grants amounting to several crores of rupees stand as a compelling testament to his visionary scientific leadership, impeccable research credential, and unwavering pursuit of innovation. His scholarly oeuvre is truly remarkable, encompassing more than 100 peer-reviewed research publications in journals of international acclaim, collectively commanding a cumulative impact factor. Furthermore, his distinguished intellectual contributions are reflected in multiple granted patents and several patent applications, underscoring his relentless pursuit of scientific discovery and technological advancement. As a dedicated mentor, Professor Subudhi has nurtured numerous PhD scholars and M.Pharma researchers, fostering a culture of intellectual curiosity, critical inquiry, scientific integrity, research excellence. His contribution has significantly enriched pharmaceutical education and has inspired an entire generations of research and academicians. Today, we are exceptionally privileged to have such a luminary in pharmaceutical sciences among us to deliver an enlightening discuss on the contemporary topic strategies for antiviral development. I am absolutely confident that his thought-provoking deliberations, profound scientific insights, and unparalleled expertise will not only broaden our intellectual horizon, but also ignite within us enduring passion for scientific innovation and translation research. It is now my singular honor and privilege to invite this distinguished scientist, celebrated academician and visionary researcher to address this August gathering. May I request everyone to join me in extending a resounding, heartfelt, and thunderous round of applause as we warmly welcome our esteemed research professor, Dr. Bharat. Thank you.
Professor Ishwari Prasad Parisa is the chairperson of this session and now the extended resources. Thank you.
Thank you, Asit. Actually, my suggestion would be just ignore his words. I came here just to be the compelling reason is that I want to thank with you all. So if you can, enhance the network of friendship, that will be the right reason for me to be here. So, respected chairperson of the session, Principal Madam, Secretary, sir, my fellow colleagues and dear friends, faculty friends and participants. So when Dr. Anjan actually invited me to give a talk on this, I thought that this topic was very appropriate because everyone is now talking of air and there is air in pharmacy. So I thought that whatever I present, I took some part of air. So what I did, I used the cloud platform is there. So what I did that, how actually, be friends with people. Only when you share your experience with them, then only become friends. So I said, fine, I will share my research experience, whatever, part of my research experience, what I have been doing with them. So what I did, I submitted a few of my papers into that platform, and I told them that this is going to be a FDP program. Participants would like to be faculty members. So give me a PPT and time hours is around 50 minutes. So give me a PPT which I can deliver. So what they give me actually 20 odd slides they gave me, and I thought there are some errors because they only give you a general data form. So if you present actually for faculty members to not be very easier. So then I started working on that. I put some slides which will be relevant to you, and that is how actually I made this presentation. So that is how from the beginning I said the introduction that I use really in my presentation, that is in preparing the draft of the presentation. So strategies for antiviral development, sir, can you? No. So what we cover, what are the disease, virus biology, little bit the targets, drug strategies, then computational AI-driven strategies if you have. Then case studies, that is our research papers briefly we will cover them, and the challenges and future direction.
I will end this.
Yeah. So the global burden of viral disease, there is no need to overemphasize it for you to all because everybody has witnessed pandemic, COVID-19, everybody has witnessed. So other than COVID, SARS-CoV-2, there are other viruses which are also, very much actually, a huge social burden to the society. But they are categorized as neglected tropical disease. By WHO, they are categorized as neglected tropical disease because they are, outbreaks are sporadic. And that is why there is not a huge market. So that is why pharmaceutical industries, they are going to invest much in them because what matters for them is commercial viability. So because antiviral market is very short, so that is why pharmaceutical industry, they actually fear to industry because product development is a very costly process. Now that AI has come into play, I think there will be more development into this field. But still, there are diseases like dengue, chikungunya, Japanese, the encephalitis, then Zika virus, even Ebola virus. They all are neglected tropical diseases. But now they are not limited to tropical regions only because their vectors are actually they have become resistant that's spreading from one region to another. So even though they are categorized as tropical diseases, they are no longer tropical and they can be found even in cold climates. And if you see that recent papers you might have seen that France, their temperature is rising around 40 degrees. Because if you are having high temperature, high humidity, there is a very good medium for virus to grow. That means what? The viruses they have grown worldwide, and they are still with this, global warming, they are supposed to grow more also. So they are going to be global warmers. So this is actually life cycle. So how actually this is the point. Can I use this? So this I will explain. This is actually related to this is related to chikungunya virus. So what we see in the first.
But pointer doesn't work with the
Okay. We do not want that.
pointer doesn't.
Okay, then let me go. So you see, there are several phases. So virus attachment will be there, then membrane fusion will be there, then endocytosis, then release is then there will be replication complex will be there, there will be the replication will be there, then virus release and mechanical. So what you find here that each of these stages, multiple things are involved. And any of these things can be okay. So whether it is your attachment, entry, fusion, replication, release, capping, everywhere you can find the target to kill the virus. That is of this one. Yeah. So each of these actually we can focus to develop actin antibody. That means here you are directly killing the virus by putting your ligand onto that particular target. So that is why we call it direct actin antibody. Okay. What is direct antiviral are expected to be very specific for the virus. Because for virus, each virus has its unique structural protein, unique non-structural proteins. No two are very much similar. There will be some metabolism they are found to be this protein and this protein. That is why if you focus on a specific target actually, so antiviral becomes very specific. Okay, so that is why we call that as direct. Then another category that is we call as indirect active. So they are to be a target. Because for replication and survival, virus is a host. So there are certain signaling pathways, certain receptors, helping replication, produce immune response. In every stage, several receptors, signaling pathways, they will all be involved. So if you can target them, and you can expect to have a broad, spectrum of action. Because there you are targeting the host. So that particular host might involve or might be responsible for multiple viruses. So if that particular pathway, suppose for example, we are in a board. For many viruses, that will be responsible for your immune reaction. So if you can target that, then you can the antiviral can be responsible. But there is a clause here that if you are targeting that post cycle, you have to because you cannot control that. It is going to stop. So you have to balance all these, you have to take consideration all this, and then design your business. Okay. So this is really how you should think of targeting or developing. Then coming to core strategies. Core strategy means here actually I am trying to say what are the classical ways in which you basically wish to develop an idea for any particular science. First is structure-based design. Purposing, enhancing solubility, permeability, then third is the natural product and hybrid molecules. Fourth is computational AI discovery. Then next is post-targeted anti-inflammatory adverts. So each case will go by example. Next. Yes. So this is actually the work of my student. So because we are actually pharmaceutical chemists, we work on product design and development. So in 2012, we made the biologist. She is Dr. Shobha from IPGA. Because she was a biologist and she was looking for something, some material to work on. So when they contacted us, we said that let us say we design it for you. So I assigned that to he developed a single molecule and got his PhD on this single molecule. So what is the beauty of the single molecule? So I explain how we develop this. So by that time we didn't have AI. Even Google was, not competent. We didn't have much resources. So then I told you that you should find some old stuff which we can read. So he stopped and found that don't take some experience. So when he stopped, so he said that some decision is one actually used against box viruses, but it is no longer used. So this is the methicillin or so this is actually certain beta with methyl substitution. So what we did that we asked for companies to give us methicillin. Unfortunately, it was not available in the market. So what we did that we synthesized methicillin in our lab. Then gave it to the biologists that you check whether it is showing any activity or not. So she said that it is not showing any activity. It is safe to use, but no activity at all. Because chikungunya was the only facility or lab. It is not effective at all. Then I said, this is the molecule should work. So we should actually derivatize that. So what we did that I told him that you find out fragments which are actually, in the literature, which are actually part of many antiviral molecules. So he screened a lot of, fragments. And then I told him that you estimate what is the synthetic feasibility because taking the fragments, developing the molecules one thing, synthetic feasibility is another. So these two things into consideration, we designed the library of almost 200 molecules. Then for the 300 molecules, this is even library because it's not available in copying or something like that. So purely 350 molecules, which are novel molecules, we designed them and screened them for product like this. So after doing that, we found that this molecule is actually perfectly, compatible. It is a small molecule, molecular weight is around 360 something some odd, drug likeness is 5, okay, and its affinity is also good for the target. Okay, the target is actually this NSP2, that is actually a multifunctional protease. That is the main protease that is necessary for CP replication, single replication. That is a very important protease, very important target. So then we did, by that we didn't have potential, we have only this, routine thing to work on. So we use automatic winner for that. And in automatic winner, we got this result. From this, we are getting minus 6.4. For our term, we are getting minus 7.1. Then what we did that all factors also is separated, and when we got there also, we found that the molecule in target, that is NAAC IPGA, is showing has activity. I said, fine, now this molecule is fine, we can work on that. So then we did molecular docking for that, and after that docking, what we found after analyzing the binding site, that it is binding to, it is binding the FDP in between two regions, that is 161013, this 151008. These are not two catalytic tags. Any protease you take, there will be certain residue will be there, which will be catalytic. They will be initiating the catalytic action of that catalytic tag. So when we found that in the vicinity of that one, actually this prediction may not be very much appropriate, but what we found that it is finding that value. Then we thought it is fine, so let us validate it. So what we did, we went for validation. So what they did is that they, take out this NSP2 protease. They compute that NSP2 protease of that particular chikungunya virus. Then they incubated our rock with them in the FDP activity. They incubated with that, and it is inhibiting the fatality activity. So how we do that? Now you put a protein to that. So if catalytic activity is there, it will break that protein. Catalytic means it will break that protein, so it will give you some fluorescence. Right? If it is not breaking that one, it will give you less fluorescence. So based on that, we found that it is actually having catalytic activity with an STP of 31.6, like, right? So this is actually very good for us because we found that initially we found that it may bind with that protease activity or in vitro validation. When we found that it is also doing the same. So now we confirm that it will be actually working against the CP. Okay, next. So then we, went from that IPGA is your inhibitory concentration. That is the concentration by which 50% virus is killed. Sensitivity is because when you are working on antiviral, you have to take, what is the toxicity to cell, toxicity to virus. Okay, so selectivity index in other terms will be giving you the benefit to this. Okay, so high selectivity is better. So we found that it is having a very high selectivity of 1. Okay, that means it is 21 times more potent against the virus than the normal cells. And it may not appear in multiple cells of it. You see that the drug was introduced at different time points. This is called, primary study. Drug was introduced at different time points, and ribavirin was the control that was used. Ribavirin actually is working on initial stages, but ribavirin was spoiled. But if you look at the drug, it is having better effect of ribavirin. Straight. Ribavirin by the standard drug is used for chikungunya virus. So these show that it is working on NSP2, it is also working on different other targets. Okay. So that is a good thing. Good thing for us that we went for in vivo validation. So for in vivo validation, we went for this active neural toxicity, OECD 423 implemented. So there we found that up to 2018 there is no death. And it happens, you classify that become with a eligibility of 2002 503 per kg. Okay. So now what we did, we thought we should be, even if the difficulty is so high, we should start with a very small dose. So what we did, we took a 15 mg per kg. That is a small dose, 15 mg per kg we did. And we did this in vivo bar. And if you look at this, in vivo bar, if you should look at this, in muscle, spleen, kidney, liver, and brain, in every tissue, MPJ and IPT was having a effect. That means it was killing the virus almost all. Chikungunya virus mainly causes itself in the muscle tissue, joints, and so on. So what we found out is killing the virus effectively. And that is how we attempted this polymer. So this is a case study, a graphical case study of how we can develop a structure-based design. That is, first of all, identify your virus target. Then here the virus target. It's not related to virus only. Any field you are on. So find out the target which is should be druggable. The target should also be druggable. Druggable means what? It can be different cavity, just like catalytic diet it has. Okay. So if it is druggable, then select that cavity and against that you do a molecular docking, find out what is the concept and the potential of that to happen, then go for validation. Okay. For you, that would be fine. That's it. Okay. So that was a classical strategy where we used structural design. Then another strategy is repurpose. So this actually we did. It is actually our invitation from it. All for DBT to submit intent for getting a drug repurpose. So when she discussed that with me, I said that there is a unique drug which you can select for this is telmisartan. Because telmisartan is H1 blocker. It is H1 blocker. And also a partial agonist of beta gamma. And if you everybody must be knowing that H1 and beta gamma, they have very interesting roles inside your system, basically in inflammation. In particular form of inflammation you take, there will be a component of H1 and beta gamma. That means H1 up regulation, beta gamma down regulation will be always associated with inflammation. Because why we did this? Because prior to this, we worked out a DRDO project. So there what we did, this losartan, losartan, its metabolite is losartan carboxylic acid, which is actually 40 times more potent than losartan. So losartan is actually a prodrug. Losartan is effective, the metabolite is 40 times more. So that metabolite can be effective against inflammation neuroprotection because in your death, the information you get, it is get a localized this renin angiotensin involved. So we targeted that with that. So we developed a prodrug of metabolite with ascorbic acid because losartan carboxylic acid does not reach brain. So we use a carrier system so that it reaches brain and ascorbic acid and LCA both synergistic effect. That is so that expense I had with that it is actually against information in the brain. So tell me certain working, certain metabolite is working, tell me also should work. Then we submitted this proposal and then approved that with the 2 years project. So there we we explore the efficacy of television circle. So I got told only about so many slides here. I will only talk whatever is relevant for you. So what we found that television circle is viable. I mean, for sale there is no here. It's up to more than 300 people. In this is in Berhampur, this is another losses we have taken. The losses also is given more than 300. And then here we are getting ICP 20, here we are getting ICP 40. So we get ICP in the range of 20 to 40 cells. So directly we screen it for inhibition. So now we have good selectivity index, almost more than 15 selectivity index is more than 15. 15 to 8 to 15. So that is good for the rock. So then, so what we did, if you look at these images, the red one is that wherever cells are there, they will stand pink. Okay, if cells are if there is minor cell is killing, the normal cells are there, you won't get that pink. So if you look at the structure, that as you go on increasing the concentrations of living cell, okay, the virus are getting killed. Okay, so then we wanted to find out why it is happening so. Unfortunately, by the time we are working on this, simultaneously there is another group was working on the same. We didn't know about it. So they published the paper, which contained single mark for that. That was from IIT Roorkee, their medical department. She is now a collaborator. So when we found that they have published this ICP-61, they have published, so we have to work more on that. So we are actually forced to work more on that because they have published that. So then we work in detail to find out what is the host axis, why it is making effect. Because they have shown that it is also inhibiting the protein. So we said that, okay, fine, they have shown what is the effect on virus. Let us say what is the effect on host also. So they didn't explore the host part. So we explored the host part. So what we did, that actually they did, I mean, Toshima Madam's lab is, they selected CW and AGP. AGP is actually, this is a angiotensin agonist. That is opposite to television. CW is actually PPR gamma and opposite to television. So by using these controls, we could see that whenever we are using AZ2 or Z2, the virus infection is shooting up. In fact, H1 is actually elevated, it is coming down, is shooting up. Okay? But if you are using telmisartan alone, virus infection is coming down. Means what? The antiviral effect is primarily mediated through these post-directed pathways. So if it is mediated through post-directed pathways, that means because earlier I told you in the beginning that we have information kind of thing, we have many a problem. It must be information. So what we did, we actually screened for many information markers. In all of them, actually we found that it is actually modulating the inflammation markers. Okay, and so it is actually this mediating this antiviral action post-directed pathways to ATR blocking, PGR activation, MAP mechanism. Okay, so these are the mechanisms we established for television. Okay. So this is how actually you can repurpose this thing. So this is what it is doing. So tell me starter is actually, normally what they use, it one is the Java both are involved. They are creating the action to copy the terms in pathway. Goes through error capability market pathways. Okay. So this is how we establish that tell me starter is effective, mediating its integral effects through multiple pathways, and it can be a very good candidate for you purpose. Okay. So how we did that, but problem is that it is intended for a particular purpose. When you are giving it for information for another purpose, then its primary indication will become as a, for example, you are giving telmisartan for an anti-hypertensive. For a non-hypertensive individual, if something comes, it will go there, it will give you telmisartan. Chances are there that they also become hypertensive. Okay. Fortunately, this is actually not part of the slide. Fortunately, when we analyze that, when we are actually giving telmisartan, the medial level is not coming down beyond the basal level. In fact, it is not reducing the medial basal level. In fact, it's not going to harm anything. So this it is not going to produce hypertension in individuals. Several studies also have established that matter. So that is how actually we are planning it clinical track actually planning for that. We are working on that. Then so as a pharmaceutical changes, you cannot be satisfied with what you get. You try to improve on. So this is our another strategy. Enhancing telmisartan efficacy by enhancing its solubility. Because telmisartan, it is a disease platform. Disease, everybody understands classification system. That means what it has good permeability, good pressure, good permeability, good solubility. So if you can increase the solubility, then in the vicinity of that one, in the microenvironment, more amount of will be available. So what is the best way to do it? You can do in surface way also, but we choose a different path because our objective was to make telmisartan effective against the encephalitis virus. That is the red disease. People support fatality rate is there, a little bit higher is there. Even if someone is not there will be a little simple area. So we thought we should be doing something so that it will be effective in Japanese and Spanish side. Then we thought of ascorbic acid. Now, why ascorbic acid? Because in the beginning I told you that we did a great activity of LCA using ascorbic acid as a carrier. So we use that not simply as a carrier, because ascorbic acid also have everyone knows that it reaches there very effectively when it is a polar drop, reaches there very effectively. And also there are several studies even in COVID, disease also nothing is there, you take up. That means what is complementary benefits are going on. So I thought we will develop something telmisartan ascorbic acid. Let us do a go. Ascorbic acid, everyone must know that it is a very sensitive chemical. Undergo appropriation is very difficult to handle. So normal methods which you do, it may not work for us. Because you have to avoid water. If you are using water, especially oxidized, you cannot do that. So what we did, we use minute amount of organic solvent. Actually not organic solvent, polar organic solvent we use. And then we use micronebulitis because other techniques didn't work. So we did a micronebulitis. So by micronebulitation, When we are doing this, we are finding that we have to put digital form out of it. I mean, it will take 15 days to form the digital. Okay. So by 15 days, we could found that some seeds are coming up, digital are developed. So this is it is forming a data with television. If you look at that, this is television, this is ascorbic acid. So this is basically this is a polar interaction. When we are coming to book crystal is not always a good way to develop solubility, because of stability issue. So what we did, then we took the DHC of that one, to look at this. Okay. So this is book crystal. This is the acceleration, this is the crystal. So what we find, we are finding a need very small endothermic or formula. Means what? The crystal is stable. There is a crystalline in the gravity is there. Also, it is a little bit amorphous because we are getting a low endothermic peaks. That is what is there. Because the crystal is stable, you can formulate it. Endothermic peak is small, means a little bit amorphous, means a little bit more. Okay. So that is also to external studies, IR, everything supported that one. We found 81-fold in enhancement in solubility. Previously also people have tried solubility of solution. They are 2, 19, 40, 20 like that. But we achieved 81-fold enhancement in saturated solubility by set plus N. That is protocol of the same after that, like our study, find out we got 81-fold enhancement in solubility. Okay. Now, saturated solubility counts for nothing if there is no dynamic solubility. In short, resolution is not good for that. Okay. Then we went for studying the dynamic solubility. So as you can see here, this is the circle. So even if you work in 120 minutes, only less than 60% is dissolved or released into the medium. But in our case, the composition is in the medium within 30. In fact, we have achieved also dynamic solubility. So having achieved a certain type of static solubility and dynamic solubility, majority of our objectives have been achieved. Then we simply did a permeability bar, but permeability is not a concept because business itself is permeable. But still we got 2-fold enhancement in permeability because of higher availability of non-active sector action as the effect of permeability. So that is the only advantage, but permeability is not an issue in that. So we achieved the objective. Then we wait for checking its viability, whether it is toxic to the cells or not. But it is only 10 seconds ascorbic acid. Both are well proven, both are APR proven. So they are not toxic at all to the tissues or the cells which we have used for viral growth. But when we read this, I think the calculation. So what we found, this is for Japanese instrumentation viral, JEB. So in case of JEB, for 10 seconds, we are getting an acidity of 12.3 microgram per liter. But for co-crystal, we are getting 6.883. So out of that 6.883, this is actually 1:1. So how we establish 1:1? Because the crystal is actually, this is for the participants actually, how you demonstrate that the crystal is 1:1. So what you do basically, you can do that in crystal. But for exactly your crystals to be bigger. So we could not get crystal exactly for that one. So what we did NMR study. In the NMR, unlike Atkinson NMR, total NMR if you take, you can quantize the area. So we did area quantitation. So we found that ascorbic acid area plus tension area is equal to co-crystal area. Means what? It is a 1:1. So if it is a 1:1 co-crystal, then the tension concentration is only 5 or 4.5 something is related to the circle, this is justification, but still it is able to defeat the 50%. So that was a very good achievement for that. Then we did sir, whether it is because of generation of the physical mixture or not. So when we compare, this is a physical mixture of generation and national deficit, but this is no different. Physical mixture is not having any enhancement efficiency. With the coefficient is that. So we, formulated the hypothesis that it is because it is empowering the generation to reach it, and because of which it is actually enhancing the efficiency. Okay. So this is how you can actually develop all these steps to improve. Here we have done for antiviral. You can do it for any purpose you want. Right? Next. Yes. Then official is fine. Then pharmaceutical changes little bit at times they become unnecessary also dirty also. So then we thought, right? So what do derivatives? Because we know for chikungunya, this is a major issue because chikungunya is not part of fatal, but polyarthritis can persist even years together, months together. So we thought, why not develop a molecular hybrid? This is a strategy 3 which you can use molecular hybrid of salicylic acid because salicylic acid will be oximeter will be there, certain will also be there. Would be there. So what we did, we developed a conjugation of that one. If anybody will be interested in pharmaceutical penicillin synthesis, you will understand that salicylic acid both hydroxy and carbon will there. So you cannot directly identify that with penicillin. There is a CH group, but you cannot directly identify because it will be competing with each other. So that is why you have to protect. So what we did, we protect the communication group, benzylation, then did esterification using scheduled esterification, and then after that we protected that one, and we got this PDI, PDI. Next. So what we found, it is, the cell viability, it is actually more safer than penicillin. Because you see, this cell viability is more than 8%, making at 800 micrograms. More safe. Then IPGA is 21 in 40.5 at velocity. More potent than penicillin. When we did this, because one object is to check it in arthritis. So again, this is a category 5, we see the category this and that category 5 compound. So when we did this, so what we found that compared to elbisatan, compared to salicylic acid plus elbisatan, this DDA we swing more effect against multiple inflammation models. Acute inflammation model that is called or then your arthritis model, CFA induced arthritis model is more. Okay, so now this is an extra, and we have got I have included that after here. So when in vivo we are incubating in liver homogenate, it is again converting to elevation in cell. So that means it is a prodrug. Telmisartan, this linkage is there, so it is a prodrug of telmisartan, which is more effective than telmisartan against chikungunya. Next. How much time? 5 minutes. Okay. 5 minutes. So then we did, this ice probe. Because for arthritis, you can take ibuprofen also. And for chikungunya, arthritis is a major issue. So we took ibuprofen as a stop. Then, just like we did in case of FDP and IBT, similarly we derivative the next. So we developed three derivatives of ibuprofen. Okay. And remember, and we got very good selectivity there for IUSS 25, IUIP 21, IUSOCA 11.9. And remember one thing, that calculation group you find in ibuprofen, it is very notorious for gastrotoxicity because of, and at the same time, it is also not necessary for box inhibition. So that is why it was a flexible position and we could modify to achieve this objective. So this also says it showed very good effect against chikungunya and against hepatitis also was acute and chronic model. Next. Next. Okay. So then this is also one interesting part. See, there are several targets which are poor protein. This is basically your envelope protein, E2 protein. If you look at this dengue, if you read that part, the dengue protease, that is also not druggable because that cavity is overlapped with something another protein, so drug cannot access that cavity. So what you do there, basically you develop allosteric sites. You develop develop inhibitors which can bind allosteric. But in case of this chikungunya, E1, E2, E3, they exist right. That means one is overlapping each other so that the cavity itself is not accessible for drug to go and bind. So what I thought, let us explore if there is any other site which you can select to for drug to go and bind that. So then we followed that pathway and we found that there is a site to which drug can bind. Then we did vertical screening and we found that Vasental is a drug which can go and inhibit if that also we validated and published that one. So where it can help you, that is finding the target identification, then vertical scanning, lead optimization, and implementation of solution. Next. 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 Yeah. So antiviral development still remains a challenge. Next. So where it is actually heading with the generative AI for real antiviral design. Why I told you that initially there was not much input. Now with the input of, with use of AI, many things are going to come up because and log setting strategy are going to be more easier with that one. So these are the strategies which can be used with the help of AI. Next. Yeah. So these are the people who work with me for this kind of work starting from 2016 up to 2025. So different time points they work with me. Next. So we worked on virus thanks to Dr. Sunataka because she is the reason I worked on virus, otherwise I would have worked on viral cancer or something else. Because you have come up with this, you have to and a collaborator who work very easily on that. And also we are thankful to DBT DSC for supporting projects. Next. And a big thank you to all.
Good afternoon, everybody. Thank you, Dr. Bharat, for your informative and explanatory lecture on antiviral drug development. Before proceeding this session, I will say that if anybody is having any question to be clarified, may please be comfortable to ask Dr. Bharat, who may clarify all your doubts. Anybody is having questions?
Okay. So, proceeding further, I wish to thank Dr. Bharat for accepting our invitation and traveling starting from early morning from Odisha and reached us in time, given time to us from debating from a busy schedule. Now, we know that we have very less chemical agents which can be used as antiviral, but scientists like Dr. Bharat who are working on this may be helpful for humankind in future to get more molecules which can be helpful in antiviral therapy. His lecture on this development on antiviral drugs may spark new minds to work on this, and they may also if they wish to, we will give his number and they may also contact if they wish to work on this. Because what I heard, he has shared his experiences, which may be interesting in some of your minds. Thank you, Dr. Bharat, for your uh, invitation assistance and your lecture. I would like to request Dr. Anjan to proceed further for the felicitation. For felicitation, I wish to invite Principal Madam Dr. Sagar Mishra, Dr. Prasad Kumar Chaudhary, Dr. Sudhir Kumar Sahu, Dr. R.S. Sarangi, and Dr. Anjan to come over the dais.
Sorry for the interruption. Professor Prasad Kumar Chaudhary sir, you need to ask one question.
Just we are running short of time and you.
No, you are free to ask. No problem. Please.
Just because this anti-retroviral is student development, I am very keenly towards observing all your presentation. Just for my personal query only, this selection of admission. Got this study for the repurposing of this for antiviral. So being a pharmaceutics person, we are more concerned about the thermokinetic changes. Administration is original form and after this was working. You tried a very novel approach of solidity. Did you observe any thermokinetic changes?
Yes. So actually, what we did, we have seen that in at simulated intestinal fluid, it is dissolving away the telmisartan within 30 minutes. That is, we know that if dissolution is within 30 minutes, that is immediate dissolution. Earlier, even 50% is not getting dissolved in standard media for that. That is a poor solution. So here solubility is high, permeability is already high, and in fact we have seen that permeability across Sweden, because we have taken Sweden of part and we have seen permeability across Sweden has increased by 2. So we are expecting that the drug availability will be more, but kinetic studies we have not done. Kinetic studies will be done actually. We are planning for a clinical talent. We are collaborating.
You have given a clue to all the formulation scientists now. They can try scope crystal development and they will get two
Provided the crystal has to be stable, because formulation kind of point of view also has to be stable.
So last screen you can go for this.
Yes.
Thank you, sir, for giving this new information.
Now it is time to felicitate our resource person. So, requested, please come on the dais and join for the felicitation. Yeah, now it is time to continue with the third session. So little bit with the program schedule. Okay, and the presentation starts with the term flip also with the teaching technique. So little flip, interchange, and thing is Dr. Patel, my teacher, who is working at Malaysia presently. He will deliver his talk online from his last 30 minutes online. He already joined. Without taking any time, I would like to introduce my teacher and the resource person for this session. And request Sudhanshu, our teaching faculty, assistant professor, to introduce his talk and continue by resource person and Srinivas Patel. Sudhanshu, please.
And honor to introduce our esteemed resource person, Dr. Srinivas Patel, sir. Dr. Srinivas Patel is currently serving as an associate professor at the School of Pharmacy, Faculty of Science and Engineering, University of Nottingham, Malaysia. He has over 25 years of experience in pharmacy education and research, having held academic and leadership positions in India, and Malaysia, including appointments of Taylor University and University Kuala Lumpur, Malaysia, where he played a key role in curriculum development and quality assurance in pharmacy education. He served as the head of the Department of Pharmaceutical Technology and programming director of the Bachelor of Pharmacy programming at Taylor University. He also serves as an honorary lecturer at Cardiff University, United Kingdom. Dr. Patro obtained his B.Pharm and PhD in pharmaceutics from Berhampur University, India, and his M.Pharm in pharmaceutical technology from University, India, where he was awarded the gold medal. His research interests include pharma, drug delivery systems, pharmaceutical technology, metal and nanoparticles, nanoemulsion, nanoforming, electrophones, nanofiber, and formulation strategies for wound healing and cancer therapy. He has supervised 15 postgraduate students and has published more than 50 reviewed journal articles and 4 book chapters. He has also presented his research and delivered invited guest lectures at numerous national and international conferences, including Malaysia, Singapore, the Philippines, Indonesia. His current interests include innovative teaching, pedagogy in pharmacy education, outcome-based education, flipped learning, and the application of digital technologies to enhance pharmaceutical education. We are very fortunate to have such distinguished academicians and researchers with us today. On the behalf of our institution, I extend a warm welcome to Dr. Srinivas Mathur, Siddhanti, and sincerely thank him for graciously accepting our invitation to deliver his today's session. Thank you, sir.
Sir, we are audible to you, sir.
Yes, audible, but
Sir,
not.
you may share your, sir. You may share your slides and start.
Let me share.
Sir, you can click on that present now option is there. Sir.
Present now. Okay.
Present now and share the entire screen.
Share the entire screen. Share the entire screen. So, okay, I have clicked the share the entire screen. Is it?
Sir, now you can open your.
Can you
Sir,
view
you
my
have
slide?
no, sir. You have to click that entire screen and I think OK option will come. Then it will be. Yes, sir. Now your screen is visible to all the audience, sir.
My slides are visible.
Yes, sir. You can go for PowerPoint show. Full screen.
Okay. Is it audible? Is it visible?
Now I think you close, sir. Your screen is not shared.
Now?
Sir, you once again you escape your full screen mode.
Okay. So,
You are audible, sir.
am I audible to you? Can you hear me?
Yes, sir. You are audible. You are audible, but your screen is not shared, sir. So just try once, or you can share your PowerPoint. We will display here. So now your screen is visible, sir. You please go for full screen mode.
Okay, full screen mode.
Yes, sir.
Present
Your
now.
slide is also now visible and you are also visible and audible.
Audible? Okay, so shall I start?
Yes, sir. Yes, sir.
Yes, sir.
Please, sir.
Thank you.
Please, sir. Continue.
Okay, actually,
Can we start?
for me, the line is not bit clear, but if my voice is clear to you, then that should be fine.
Yes, sir. You are audible. Completely clear, sir.
If in the middle, if there is any issue or problem, then you can always let me know.
Sir.
Okay.
Please continue, sir.
Okay. So first of all, thank you very much, the moderator, for the nice introduction, and I thank Dr. Anjan and also Royal College of Pharmacy and Health Sciences for giving me this opportunity to present a talk in this esteemed faculty development program. Okay, so let me start. So when Dr. Anjan approached me to deliver talk on teaching pedagogy, I thought of few topics, and I think that this flipped learning concept will be a good one to go. But later, when I received the invitation letter, in that invitation letter, there is another sub theme like teaching pedagogy aligned with the National Education Policy NEP 2020. So that has triggered me to go into details, like let me have a better understanding of what is national education policy and how I can align my today's topic to the national education policy. So I have gone through the document, National Education Policy, and the PCI BFOM syllabus, and I have extracted few important points which are relevant to my talk, and I have integrated. So hope it will be useful and I can justify to a certain extent. Right? Okay. So once again, thank you for the opportunity. So let me start. Okay. So as I understand that this is a faculty development program, and many of you are from different disciplines. So my topic today is focusing on one of the practical aspect of pharmaceutics. But still, just I want to know that how many of you belong to pharmaceutics and how many of you are from other branches. Okay. So can I request all of you to open your phone and open any browser and type join class point, and when you join it, you will be prompted to enter a code, and then you can enter this class code 81260. Okay, so just to keep the ball rolling, let me see that whether this app works or not. So can you open your phone and open any browser like Google and just type join class point and enter this class code. And you can just answer which is your or what is your primary teaching discipline, so that I will just get an idea that how many of you are from each discipline. Just give like another 30 seconds and please try to answer this.
Only join the class point to add for thousand. Not required to download. You can just click on the first link. Put this code 8126.
Okay, anyway, so thank you very much for your participating. I'm closing the submission now in the interest of time. Okay. So what I got is that maximum of you are from pharmaceutics, and also some of you are from other branches like pharmacology and pharmaceutical analysis. Okay, so that's great. So though my talk will be mostly focused towards the one of the pharmaceutics practical, as I mentioned in my title, it is a flipped learning approach for pharmaceutics practical. But once the principles are understood, that can be applied to other practicals also. Okay, so with this, let us proceed. Okay, so before I proceed, a brief information about the university where I am working currently. So the name of university is University of Nottingham, and happy to share that this is in the top 100 universities in the globe. Okay, this is world rank. We are 97, and recently last year we have celebrated 100 years of School of Pharmacy at University of Nottingham, UK. Okay, as well in Malaysia campus we have celebrated 20 years. But this is the overall ranking. But in pharmacy and pharmacology category, we are ranked 8 in, in the world, top 8. Okay, so we have entered recently into the top 10 category, and this university has got many reputations to its credit. Okay, so one of the notable reputation is so the discovery of the most popular drug ibuprofen by Dr. Stewart Adams was held at Nottingham actually. Okay, so he was a Nottingham alumni, and in collaboration with Boots Pure Drug Company, ibuprofen was discovered in the early 1960s. So this is one of the notable reputation, but there are many others to its credit. Okay. So coming to the outline of my presentation, so I will start with evolving expectations of the graduates. Okay, expectations means expectations by employers, by the regulatory bodies, etc. Okay, regulatory bodies, I mean PCI, Pharmacy Council of India. So then I will be just touching what are the requirements of PCI according to NEP 2020, and some aspects of outcome based education and Bloom's taxonomy. And later I will highlight what is flipped learning. Okay, we'll see what is flipped learning and how it can be applied to our teaching. Then I will specifically go through a case study, phenylephrine injection BP. Okay, how to prepare an injection in our lab. Okay, and finally I will conclude with some of the opportunities and challenges in implementation of this flipped learning approach. Okay, so coming to the employers' expectations. So if you see, this is statistics according to National Association of Colleges of Employers. So if you see over a span of, hello?
Yes, sir, you are audible. Please,
Okay,
please continue.
okay, so can I proceed?
Yes, sir. Please continue.
Okay. Okay, so this is the screening of candidates by the employers. Okay, so if you see this in like around 10 years back, 2016-17, employers were giving an importance of 70% to the GPA, okay, the score. But as with time progresses, the percentage of importance given to the GPA is declining. Okay, now if you see the latest figure, like around 30% importance is only given to the GPA or GPR percentage. Okay, so but nowadays, mostly the employers are looking for skills, okay, rather than this percentage, okay, or the GPA. Okay, and if you see this graph, so this tells that more than 88% of the employers look for problem solving skills, and then ability to work in a team, okay, and followed by communication skills, initiative, etc. Okay, so the most important skill that is required or that is seen by the employers is problem solving skills. So we have to see that whether the graduates which we are producing are possessing these skills or not. Okay, so this is a general survey, and then I also gone through the latest PCI syllabus that was designed as per NEP 2020, and this revised PCI syllabus places a greater emphasis on outcome-based education. Okay, outcome-based education, student-centered learning, and development of higher order thinking skills. Okay, so we will go through some of the aspects, and later we will check whether what we are teaching are producing students of higher order thinking skills or not. Okay, so maybe let us, as I have told you that PCI syllabus emphasis on outcome-based education. May I ask you another one more question? How familiar are you with the outcome-based education? Okay, so can you open the same app, enter the class code, and try to answer this question? How familiar are you with the outcome-based education? Okay, good. Okay, so if I see the poll, it is very good to know that most of you are familiar with the outcome-based education. Okay, so very interesting. Okay, so I close the submission now. The interest of time. Okay. So that gives me idea that I should not go deep into the outcome-based education. I can just superficially go through the slides and just go to my flipped learning concept. Okay. So just, yeah, very quickly I will be going through the slides. So what is OBE? So OBE is not what we want to teach as teachers. Okay. So like in the traditional teaching method, we always focus on the content delivery and we think that whatever we teach with whatever we want we will teach. Okay, but the trend is shifting. So OBE states that it is what the students should learn, so the focus should be changed to the students what they should learn, and accordingly we have to adapt and change our teaching methodology. Okay, so few slides from now onwards, like 3 to 4 slides, all are these copied from the PCI syllabus. Okay, so it is nothing from my side, just to say you that what is outcome based education. Okay, so outcome based education is an educational theory that bases each part of an educational system around goals or outcomes. So always we have to deliver based on the outcomes that we think that the students should get. And what is an outcome? An outcome is what the students should be able to do. Okay, so the do factor is most important, whether the students can do something or not. If they can do, then you can say that okay, they have achieved. Okay, but if we focus on ourselves, what we teach, that is not enough actually. Okay, so outcome based education revolving around these levels. Okay, so like PEOs, POs and COs. Okay, so you may be knowing that PEOs, I'm not going through the details. PEOs are the objectives which can be attained after 3 to 5 years of graduation. POs, program learning outcomes, these should be achieved at the end of the program after they pass out the grad B form. And course outcomes should be achieved at the end of the completion of each course, like at the end of each semester. Okay, so these are the PEOs stated in the PCI syllabus. So if we see these PEOs, actually PEOs, so this is the suggestion of the statements by the PCI. It is not that we have to follow as it is. We can change the PEOs as per our institutional or university or college vision and mission. Okay, what is our vision, what is our mission, what type of graduates we want to produce, accordingly we can change the PEOs. But these are the very generic PEOs which are suggested by PCI, but we can take it as a guide. Okay, so if we see the PEO 2 and PEO 3 highlights that the students should demonstrate practical skills. PEO 3 highlights use scientific reasoning and problem solving skills. Okay, so whenever we are producing any graduates, we have to make sure that they should be able to demonstrate practical skills and problem solving skills. Similarly, these are the POs, program learning outcomes. So there are 11 program learning outcomes suggested by the PCI. If you see point 2, 3 and 11, same thing, they highlight they should have problem solving, critical thinking, okay, and pharmaceutical skills and practice. And finally PO 11 talks about lifelong learning. Okay, so we should be able to produce a graduate who has the ability to learn by himself or herself. Okay, so that is called as lifelong learning. Okay, so PCI also tells that we want to produce graduates with certain attributes. Okay, so here 11 attributes are given. Out of that, few attributes are highlighted: scientific and analytical thinking, industry and practice readiness, innovation and problem solving mindset, again lifelong orientation. Okay, so why I have highlighted all these things, I want to stress that these are the attributes, these are the skills which PCI wants us to develop in the students or graduates which we are producing. Okay, so that is one thing that we have gone through, the description or the definition of different types of PEOs and PLOs and COs. Okay, but how we will ensure that the course learning outcome or COs are attained? Okay, again this is a definition. given by the PCI. Okay, CEOs are attained by, it should be attained by all students at the end of a formal course, because it's a course outcome. At the end of a formal course, they should obtain. And while the method of computation of attainment of CEO is not unique, okay, they have not stated how you should adopt a method to attain the COs. So it is the responsibility of the each institution. Each institution has to follow a well-defined approach, whether it's a direct approach or indirect approach, and make sure that those CEOs are obtained at the end of the each course. Okay, so this is very important. A foundation of OBE is called as constructive alignment. Okay, so whenever we are designing a syllabus, okay, so we have to come up with the learning outcomes. Okay, those learning outcomes are based, as I mentioned earlier, based on the program educational objectives, program learning outcomes, and course learning outcomes. Okay, so we have to design those learning outcomes. Then we have to think that how we can deliver, how we can design our teaching activities, teaching and learning activities, so that these learning outcomes are obtained. Okay, so what can be the teaching and learning activities? The general pedagogical techniques like lectures, practicals, and other activities like presentations, assignments, problem-based learning, case-based learning activities, etc. Okay, and once all these things are delivered, the most important aspect is the assessment. We have to assess that whether those learning outcomes are obtained or not, or achieved or not, or attained or not. Okay, so then if you do an assessment, and then if you go for, so here LOs are designed, then here T&L activities are designed, then they are delivered, and then assessments are done. And then if any improvement is needed, we have to review the module, how it has gone last semester or last year, and what are the areas of improvement and what we can do to further improve. Then again, you go back to the learning outcomes, you have to revise, okay, if needed at the end of 4 years. Okay, then you do a curriculum review, and again devise the new teaching and learning methods and do assessment and continue. Okay, so this is called a CQI, continuous quality improvement we have to do to ensure that all the COs and PLOs and PEOs can and are obtained. Okay, and how? So I told that the assessment is a one important component. So how you can assess? Okay, for proper assessment, we can follow Bloom's taxonomy. Okay, so Bloom's taxonomy is a hierarchical framework used to classify the educational learning outcomes. Okay, into levels of complexity and specificity. Okay, what type of skills we want the graduates to obtain? Okay, so those skills range from lower order thinking skills to higher order thinking skills. What are lower order thinking skills? You can go to the base of the pyramid, like remember. Okay, if they can retain and recall the information what they have learned, then you can say that they have achieved that. Okay, then if they can grasp the meaning and explain something, that is called as understanding. And this remembering and understanding are considered, and sometimes up to apply. Okay, can they apply the knowledge which they have gathered initially into a new context? Okay, this is called as application. So these are all classified like up to lower order thinking skills. But with that knowledge, can they analyze, explore relationship between different categories? Can they make judgments based on sound analysis? And finally, can they use all this information to create something new? Okay, so this levels in the upper pyramid will provide that a clue that they got higher order thinking skills. Okay, so we can use this pyramid to design assessment, and we say that so there are, for each level there are certain verbs which can be used during the assessment. So for a simple example, I am telling that suppose if you tell that you want to produce a graduate who with efficient communication skills. Okay, so when we say that communication skills are to be achieved, then we have to test it also, assess that whether those communication skills are achieved or not. So how you can assess? Then you should have a presentation as a form of assessment in your module. If you do a individual presentation or group presentation and the student present and pass that component of the assessment, then we can say that yes, the student has passed, and we can say that that communication skills has been obtained. So similarly, a problem, so problem based skills means you have to design some problems and give to the students and see that whether they are able to solve that problem or not. Okay, so that is problem based skills. Similarly, many other skills need to be assessed. With this brief introduction, let us ponder to a critical question. So if our course outcomes require students to analyze, evaluate, and design, so these are like little bit higher order thinking skills. Now coming to the practicals, are our current practical teaching and assessment methods really measure these abilities? We have to see, right? Okay, so this brings us to the next point, that we have to design our practicals in such a manner that it will develop the problem solving skills. Okay, so can I just go through one more question, because my focus is practical class, teaching practicals. So either it is pharmaceutical practical or any other subject, practical from any other subjects. Can you tell me, like just in one word or 2 to 3 words, when a student enters to a practical class, what do they usually do? How the practical class is started? Okay, so here you need to type your response. What is the first thing students usually do when they enter a pharmaceutics practical class or any other practical class? Or what is the first activity they do in the lab? Or as a lecturer, how you are starting the practical? Okay, good. Okay, so they follow the standard operating procedures, SOP, I think. They wear the apron, very good. They have to follow those things. But I'm talking about the real practical. What is the first activity? Arrange the requirements. Okay. Go through the manual. They simply start gossiping. Yeah, that is usual. Know about the instruments. Know the aim of the experiment. Get demonstration of the experiment. Okay, consult with the professor. Okay, very good. Okay, so I got some of your important points, like go through the manual, set up the equipments, know the aim of the experiments, get demonstration of the experiment. Okay. Okay, thank you very much for your participating. So I just close the submission. Okay, thank you very much. Right, so this is what we think and we usually observe when students come to practical. Okay, so some of the important takeaways are go through the manual, okay, and set up the experiments, okay, get the requirements done, okay, all those things. Right, okay, so if we think like that, I think those are considered as traditional practicals. Okay, traditional practicals. So one of the important point I got is go through the manual. Go through the manual means the manual is provided to them and they will go through the manual in the class. I understand actually. I understand like this. Or so that is more towards traditional practicals. Okay. But those traditional practicals, we have to think that can that achieve or can that lead to developing higher order thinking, okay, or higher order learning. Okay. So this brings us to a concept called as flipped learning model. Okay, so this is the heart of my talk actually. So flipped learning model. So what is flipped learning? So if we see on the left side, it is traditional model. Traditional model mostly it is teacher centered. Okay, so there are some of the activities which happens inside the class and some activities happens outside the class. Okay, so inside the class, what happens? That is the teacher's effort. So teachers demonstrates, okay, as one of you told that demonstration of the experiment. Okay, so when the teacher demonstrates, the students listens to the teacher and carry out the practical, and then maybe outside the class they go and maybe write the report and submit. Okay, analyze the results, write the report and submit. Okay, so that is happening outside the class and that effort is students effort, and this is called as this usually happens in the traditional model. Okay, so in the class what happens, knowledge and understanding aspects of the Bloom's taxonomy pyramid happens, whereas outside the class the higher orders is achieved. But because it is the student's effort, normally it will be not efficient. Okay, but if we flip the model as the name suggests, what we do is that the lower order thinking like knowledge and understanding, let them do outside the class. Okay, so that is the student's effort, and once they enter into the class, then let us collectively do some activities which will prompt or to them to achieve the higher order thinking. Okay, so this is student centered or flipped model. Okay, so the name, as the name tells, flipped, it is just flipping the activities. Okay, instead of the doing the main the core activities or the lots, when I say lots, lower order thinking skills in the class and hots in the outside, it is better to do the lots outside and come to the class and do the hots, so that collectively we can say that the skills are achieved. Okay, so then what can be the difference between the traditional and flipped practical? So in the pharmaceutics point of view, I am telling that in a traditional practical, what happens, we give a formula to the student. Okay, that you prepare suppose so and so solution or emulsion something, and then the procedure is explained, then the students follow that procedure and prepare the product. Okay, so all these things is called as teacher centered approach. Okay, but in case of flipped approach, what we do is that we will not give the formula. What we do is just give a problem, then outside the class, before students come to the class, let the students do some homework and do some uh, literature searching and come up with a formula. Okay, so that is students prepare something beforehand. Okay, where some thinking goes behind. Okay, then that is called as formulation thinking, and then with the formula they come to the class and then there will be a discussion. Okay, and once that is approved, then they go for the preparation. So then this approach we can call it as a student centered. Okay, so with this I just go quickly through a case study how to run a practical using a flip learning approach. Okay, so I have told you that we can give a formula, a problem. Okay, what is the problem? Suppose I tell to the students, this is the practical, aim of the practical, prepare 10 times 2 ml of ampules of phenylephrine injection BP containing 10 mg per ml of phenylephrine hydrochloride. So only this is the question. Okay, and they have to come up with a formula. Okay, so where do we start here? We only know the name of the drug and how much drug should be present. That's all. Okay, but you have to prepare, because in injection you can think that it is just mostly a solution. Okay, but how to move ahead. Okay, so another question, maybe again you can open the class point and you can think that you can ask yourself what formulation decisions need to be made before preparing this product. Okay, in other words, I want to say that what other ingredients that need to be present inside this preparation, apart from the drug. Phenylephrine is the drug. Now you want to prepare a solution which is to be injected, okay, because these are ampules. So what other formulas, excipients need to be present? Indirectly, I am asking what other excipients need to be present in the solution. Okay, good. Vehicle. If you want, you can specify the vehicle. What can be the vehicle? Prepare isotonic solution. Very good. What other ingredients are needed to prepare this product? Sterilization, very important. Solubilize the drug in the vehicle. Very good. If the drug is not soluble, you need to solubilize. Adjust the pH. Yes, if needed, you have to adjust the pH. Add an antioxidant. Yes. Okay. Okay. Okay, okay, very good. Thank you. Thank you very much for your participation. So I close this submission. Okay, so yes, these things have to be taken into consideration while preparing a solution for a injectable. Okay, so still, as some of you have told that you have to adjust the isotonicity, but we also have to think that do we always need to adjust the isotonicity, and if not, then in which cases we have to adjust and which cases we need not adjust. Then some of you told the sterilization. Okay, there are different methods of sterilization, like terminal sterilization and filtration. So which method you should go for? Some of you told that we need to add preservatives. Okay, if you need to add preservative, which preservative you need to add and why? Also, you have to think that do we need antioxidants? Yes, someone of you have told that you have to add antioxidants. Also, we have to think that what does the BP or the official books say about it, about the stability? Okay. Also, we have to take into some considerations like to account for the losses. Do we need to add any excess or overage? And also, what about some packaging and labeling? Okay, so these are some of the aspects we will tell to the students that your approach should be like this. In which case isotonicity is required and which case it is not. Which case sterilization is required, and if sterilization, which sterilization you have to go, terminal sterilization or filtration. Whether you need to add a preservative or not. If you need to add a preservative, which preservative you have to add. Similarly, antioxidant, filter, there are different types of filter like 0.45 micron filter, 0.22 micron filter, etc. So we will be giving all this briefing as a pre-class learning activity, and then they have to take this basic information, and then when they get the problem, they have to utilize that basic information and do a homework and come up with a formula. Okay, once the formula is developed, with that formula they will come to the lab. Okay, and once that formula is presented to the lecturer, then there will be a discussion between the lecturer and the students, and once the lecturer approves the formula, then the students goes and develop the product. Okay, so this is what happens. Okay, so very briefly, I will take 5 to 10 minutes to just tell you that what are the basis of adding these formulas in excipients. Okay, so first of all, excess and average. I know that you may be aware that what is an excess. Excess means we need to add certain amount of the product into the container. Suppose if it is 2 ml, then you can add another 10%. Okay, sorry, that is overage, another 10%. Overage means additionally you will be adding some amount. Suppose if it is a 2 ml product, you can add 2.2 ml into the ampule, so that, because if you have to draw 2 ml, normally we cannot, when you draw with a needle, you cannot draw the whole amount. So to compensate that loss, we have to add some extra amount. That is called as overage. Whereas excess is you need to account for the manufacturing losses during the preparation. Around 30 to 50% you can make. Okay. Sterilization, method of sterilization, I have told that there are two methods of sterilization. So you have to see the pharmacopoeia or any other official book, whatever is mentioned. Suppose if you see here, for British pharmacopoeia states that phenylephrine injection, the solution is sterilized by filtration. Okay, so because there is an official guidance that you have to go for filtration, then you should go for filtration. You should not go for autoclaving or terminal sterilization. Similarly, isotonicity. In which cases you need to adjust the isotonicity. If it is a aqueous preparation meant for either subcutaneous, intradermal, intramuscular, or if it is a large volume IV intravenous parenteral. Okay. Suppose if it is a small volume, suppose if it is single dose small volume, tonicity is not required actually. So tonicity is required only in cases of subcutaneous intradermal. Okay, I'll not go through the details. Okay, just this is for your idea. Okay, so here it is mentioned that in the BP, phenylephrine, usual dose phenylephrine hydrochloride by subcutaneous or intramuscular injection. So because it is mentioned, it can be administered by subcutaneous intramuscular injection. That means we need to adjust the isotonicity. And how to adjust the isotonicity? You can go by any of the popular methods like freezing point depression method, etc., and you can calculate the amount of suppose a tonicity adjuster. Here you can use sodium chloride as a tonicity adjuster. So you can use a freezing point depression formula, W is equal to 0.5 to minus A by B, and incorporate the values and calculate how much sodium chloride is needed. Similarly, preservatives. Do we need to add preservatives to every formulation? No need actually. Okay, so suppose some cases, if a preservative itself has adequate antimicrobial property, no need. Okay, but if your mostly preparations are aqueous preparations, you need to add. Okay, and in some cases, like if it is an intraspinal or epidural injection, you should not add any preservatives. Okay, also if it is a single dose and terminally sterilized product, we should not add preservatives, or we need not add preservatives. But if it is by filtration or aseptic filling, then we need. And again, for large volume parenterals, which are where the dose exceeds, or where the volume exceeds more than 15 ml, also no need to add any preservatives. Okay, so there are some regions why we need and why we not need, but here just I'm saying that these are the things which we tell to our students. Okay, and if preservative is needed, which preservative you have to add, you can follow the official books. Like this is a reference table obtained from Pharmaceutical Codex. So this is a list of preservatives you can use it, but again you have to check the compatibility of these preservatives with the active ingredients in different books. Like you can refer to the Martindale, etc., and you see that whether these substances are compatible with the drug or not. If it is not compatible, then only you can add. Okay, so here it is already mentioned that here the solution is sterilized by filtration. If filtration, definitely you need to add a preservative. So you can go and add chlorocresol, which is a commonly used preservatives in parenterals. Okay, similarly antioxidants. No, here in the British Pharmacopoeia monograph for phenylephrine, there is no mention of antioxidant. That means no need, because if it is needed, then definitely they will mention. Okay, so if it is not needed, you need not mention. But if it is needed, suppose there is another example like procainamide injection. So it is written somewhere here that you need to add an equivalent of 0.1% sulfur dioxide, so you can use it. Okay, so based on the above information, so we see that antioxidants are not required. And if you take all the excess and overage into consideration and you round it for easy calculation to 40 ml, so if the total volume need to be prepared is 40 ml, so this much will be the 400 mg will be the phenylephrine, chlorocresol around 20 ml, sodium chloride you have used the freezing point depression method to calculate, and you. So this formula the students need to do before coming to the class. Okay, so here I have, this is my formula, but the students may come up with a different formula also. They may use a different preservative, they may use a different tonicity adjusting agents, like that. So but we can discuss in the beginning of the class that whether their formula is acceptable or not, and whether they can use that formula. Sometimes they can suggest another preservative, but that preservative may not be available in the lab, so we can tell that okay, you are correct, but we cannot use it. We can use the another preservative, like that sodium chloride. Suppose they tell that we can use potassium chloride, but potassium chloride is not available in the lab, then we can tell that instead of potassium chloride we can use the sodium chloride, like that. Okay, so this when they do all these activities at home, then that will be creating the critical thinking skills and problem solving skills once. Okay, so apart from that, we also have to take into consideration some additional pre-laboratory consideration. They also have to look into other aspects. Okay, so I am not going through these details. Okay, so what happens, I have told you in the practical. So students present the formulation in the beginning of the lab, then they discuss the rationale, they receive the feedback from the lecturer. If needed, the formula is modified, and finally the product is prepared. Okay, and finally they have to come up with the label. Okay, so I'm not going through. Okay, so with this we are coming to an end. So these are the opportunities and challenges in implementation. So the opportunities include better student preparation if we follow a flipped learning approach, or higher engagement. It promotes active learning, critical thinking, problem solving, and finally self-directed learning, which is most important for getting lifelong learning skills. But the challenge is that mostly there is a student resistance, because the students need to do additional work. Sometimes they will be not cooperative, so we have to motivate them. And also for the staff, you need additional preparation time to come up with the problem, and also you have to check that whether adequate resources are available or not. Okay, so if we bring the Bloom's taxonomy pyramid here once again, so if we see at the remember, we say that okay, the formula is recalled. Okay, so they can remember. Then if they explain the rationale, then we can say that yes, they understood. If they can able to do all the pharmaceutical calculations, then we can say that yes, they could apply. Then compare the formulation options available, then you can say yes, they can analyze. Then justify the excipient selection, then they can evaluate, and finally they come up with the working formula, and so you can say that they have created. So we have achieved all the higher order thinking skills also by flipping the practical. Okay, so as I have told you, this is one of the case study which I have told. But when I tell you, you implement these things, sometimes it may be difficult to implement entirely, but you can take it as a cue and start with at least one practical. Maybe you can design some pre-lab worksheets, let them do some work before coming to the class, or a pre-lab quiz also you can do, so that the related concepts you can create, take into consideration and create a small quiz. Okay, and once they come to the practical, they do some homework and come, so that you can get a platform to discuss and provide feedback. Okay, so with this we can slowly start implementing and ensure that the higher order thinking skills are obtained. Okay, so this is one of the aspect of ampule I have told, but you can also apply to ophthalmic preparations or semi-solid suspensions, emulsions, tablets, capsules. Not only pharmaceutics practical, these same concepts can be taken, and you can adapt to your own practicals, whatever discipline, and you can develop these skills. Okay, so to conclude, so what we have seen, employers and the new PCI curriculum emphasizes graduates with problem solving and higher order thinking skills. And to obtain this, one of the good approach is flipped learning. Okay, so flipped learning provides practical strategy to promote active learning and higher order thinking in pharmaceutics practical. Okay, so if we start with small changes in practical teaching, it can lead to meaningful improvement in students' learning. Okay, so students should not simply prepare formulations like traditional practicals. They should learn to think like formulators. Okay, so finally what I say is that don't give the students directly the formula, give them the formulation problem and let them solve by themselves and develop their skills. So with this, thank you very much for the opportunity, and if time permits, then I'm happy to take a couple of questions. Once again, thank you organizers for giving me this opportunity to share my share the talk actually on flip learning.
Thank you so very much, sir, for a very simple and beautiful presentation. I hope all the participants might have gained something important from your presentation. That's so good and nice. Now it is time and the session is open. Faculty to ask any questions or queries from your presentation. Now it's open to all.
Yes, please.
Yes, audience, please. Many pharmaceutical personnel are there. Yeah, I will ask one thing, sir. The important one is what you highlight students' resistance. So
Sorry,
now
sorry, Anjan. Actually,
want to go in.
Anjan, the line is not clear to me. I am not able to hear you clearly. If any specific question is there, can you write in the chat? I can read and answer. I cannot hear you clearly.
Excuse me, sir. Am I audible to you?
Yeah, now audible. Now audible.
Am I audible to you, sir?
Yes, audible.
So, just simple question. It's one interaction only, no question. So,
Yes.
how is the students' learning attitude there in Malaysia? And you can just, as you are from India, and you have teach many years here in Berhampur and in Odisha, and how you feel the difference with learning attitude, the students in Odisha or India, and the students' learning attitude at Malaysia or abroad, as per your experience. One thing. Second, yes, what all are human beings, so mostly all have the same human thought. So what you will tell, and there also the students will have the same issue of the resistance to work with their own. So how you motivate or encourage, and how the organization follows that. The same thing. Just two questions. What is the difference between the learning attitude of students here in India and abroad at Malaysia, like that, and how you motivate? Simple, just one interaction I need to hear from you.
Okay.
Am I clear, sir?
Thank you. Yes, you're
Learning
clear, Anjan.
attitude and your activity you follow there.
Thank you very much for the question. So you see, we cannot blame the students, because all the students' attitudes depend upon the environment, the scenario, the institute, the university, the staff, everything. Okay, so how the students have been grown up, what they have seen around them, they will behave accordingly. Okay, so if you teach students, if what we usually teach students since beginning, from the school days, from the college, from the foundation level, so they will think that this is how the education goes. But in Malaysia, what happens, the education is bit student oriented right from the schooling itself. So always in the schools, all the activities are based on problem based activities. They are giving always the tasks. They will be always given assignments to do at home. They will come up and share their experiences within the class. So they grow up like that. So because they grow up, the resistance of those students will be minimum here. But in India, suppose in India or anywhere, if we suddenly expose the students to certain new aspects, definitely the initial resistance will be there. But if with the time, if we do a bit of efforts and we train the students, we motivate the students, then students will be definitely changing. Okay, so my answer is we should not put the blame to the students. Students are students. How we train them is more important. Okay, so here we develop skills, we learn new things, we try to implement slowly. Over a period of time, we can change the system. Have I answered your question?
Yes, yes, yes. You very aptly you answered the question. I know you, and your answer is also as clever as you, sir. It's a brilliant answer only, not simple answer.
It is not
Okay,
clever.
definitely we have to
We have to
motivate.
see the
Not
reality.
our job, the most important job of a teacher is to motivate the students. Just not only
Yes.
teaching what is written in the book. So creating
Yeah,
interest
true.
is the major role of a teacher part of in the life of a student. Thank you so very much, sir. So now it is time. It's online only, so we cannot felicitate you here. But at least this I will keep with you or in our office. When you will visit to Berhampur, so when you will visit to Berhampur, so it will be our pleasure to invite to our organization and our kind request to deliver a presentation physically here also and to honor you with our most respect on behalf of Royal College of Pharmacy and Health Sciences. So thank you so very much, sir, for giving your very valuable time to we all, and thank you once again on behalf of all the members of this Royal College of Pharmacy and Health Sciences, and on behalf of the entire program, which is recognized by Pharmacy Council of India, PCI, New Delhi, and the association with the IPGA Odisha branch. Thank you so very much, sir. We are so much honored to get you and listening from you.
Thank you very much,
Thank you.
Dr. Anjan.
So with this, we can stop this session.
Yeah. Thank you very much, Dr. Anjan and the entire organizing team for also giving me this opportunity to share my ideas. And I wish very all the best to the successful completion of your faculty development program. Thank you very much, and have a good afternoon to all of you.
Thank you. The session is over now, sir.
Okay.
Now time to go to the next session. Medical coding and the members are basically and I like to request and invite the team, Mr. Anumati Chaudhary, Mr. Bala Murali, and Madam Veena to please come on the time. And I request for a welcome to all the team members from the team organization. It's time to introduce the organization and the members. And I request Dr. K. Harikrishnan to please come on and introduce the organization and the team members. After that, we'll continue the session. Thank you. Take your seat.
Professor at College of Pharmacy. Now, Mr. Balamurali Krishna, Director of Hospitals Medical Coding Academy, and Ms. Dhanmati, Founder and CEO of Hospitals Medical Coding Academy. Hospitals Medical Coding Academy has been started in the year 2016 with one sector and one branch. Now it has expanded to around 12 branches in 4 different states, and there are around 25,000 students in this decade, and they have provided assistance to around 25,000 students during this period. And they have been nominated for several international and national awards, and like the Excellence in Medical Coding Academy Award in Abu Dhabi, and the award for Excellence in Medical Coding Training and Development nomination for the Radio City Awards. Now, I will the speaker, Mr. Balu.
Good afternoon. Very happy to be part of this. Thanks to Royal College of Pharmacy. So, because I'm from pharmacy background, I studied my pharmacy. And I recall my college days because after the Barista and Steve Sir sessions, I forgot in my main year, I said in the first one, I'm a student and visiting the session. Okay, how the college will be like that. So really very happy for that. So you recall my pharmacy day sessions. So happy for that. And as a pharmacy graduates, even in our college also, every time they used to call this so many guest lectures and all. So always I should say in the first one, listen the sessions. Whether it is useful or not useful for me. I believe that it one thing it will be helpful for me. So like that, all the sections. After my graduation, I worked in different pharmacy companies, hospitals, I work, agencies everywhere. Then I choose the career as medical coding. So I worked with the MNC companies in Chennai and Bangalore. Then I said, pharmacy itself, what it will be, I want to start my own company and be more employment in the state and on. So that's how my journey got started. Then currently, we expanded into multiple business. We are running a 10 business with the training and our own production. So we are making coding projects also. And now our company is like multi-million company. I'm very happy for that. So there is a lot of opportunity not only confined to the pharmacy because we need to educate the students, right? So that's our responsibility as a teacher. So if you educate properly, definitely I think they come back to the same college and they can take this lecture. So that's only which I have. So in today's session, I will take about the complete part of the medical coding, how the growth will be there. So that will be helpful for you to educate the upcoming pharmacy students. So we are okay with we'll go to the session. I think maybe finance break and we can go ahead. So I want to introduce my partner with me. So we'll go together to take this session.
Hello everyone. Very warm morning. So all are so that's a huge break. Yes. So can we complete which is right into alphanumerical or numerical codes. This is what the medical coding complete meant for.
How we do coding. So there are three books we are using for this medical coding process. The first book is called as IPGA, which stands for International Classification of Diseases. Okay. Every year there is a new disease will come into picture, right? Each and every disease will updated in this book by WHO, World Health Organization. So recently we have COVID disease and related disease condition, right? So everything we can see the new codes in this. So this book was published by WHO. And the second book is called CPT, which stands for Climate Procedure Technology. That means any surgery, whatever the surgery, maybe a suture removal or suturing or connectivity or open heart surgeries, all surgeries begin available in the CPT. The last book is a HCPS book, which is the Healthcare Common Procedure Coding System. That's from pharmacy background, this will see all the common drugs injection, right? Each and every drug, whatever we do formulation, everything was printed in this third, which is called HCPS. So what as a medical coder we do is, so we will read the medical records. So from the medical record, we convert all the diagnosis, procedures, and supplies into codes. Again, every year these books will be updated.
So what is ICD?
International classification of
International classification of diseases. That means in a patient body, what went wrong? That means what is the disease? That will be coded from ICD. And for the disease, what the treatment has been given. Either it may be a surgery or a procedure. We are going to convert from CPT. CPT means procedural terminology. And for providing the services, that means surgery or procedures, whatever the supplies, either cotton or a thread, we are going to convert from HCPCS book, Healthcare Common Procedural Coding System. So all the three books we are, that means the manuals we are going to use for medical coding. So now he said that all the codes will be available in the manuals. ICD contains more than 74,000 of the codes. CPT book contains more than 10,000 surgeries in that. Is for any human, is that possible to memorize? No, we can't memorize the codes, so that we have separate options.
So instead of manual, because for a different examination, so whenever we want to work in this domain, we have to get certification from US, UK, and Australian certification certificate. For examination purpose, only we use a course. But companies, they use the software. By using the software only, we will take all the whatever this is condition you want to put for the case study.
So there will be one patient medical record, several departments will work for that. That means patient will be visiting the hospital. Before meeting the doctor, he should meet the reception where the trials will be taken. That means the weight, BP and all. After that, he will meet the doctor. If doctor orders him any lab or radiology sessions, the X-rays and all, he need to go there. If he is having a severe condition, he need to get admitted in the hospital. So, how many departments we are seeing? There is an outpatient, laboratory, radiology, inpatient, like that. For one patient medical record, many departments will work. All the departments have separate set of coding.
So coming for the coding, if you are not doing proper coding, it will have a huge impact on the hospitals. Because currently we are not doing coding in India. We work for US projects, DA projects, other country projects. Okay. So the main thing, if you are not able to code properly, the claim will get rejected, and it will lost for the physician. A lot of money is needed. So if you have proper coding, so if you have the accurate claim submission, the faster payment your physician will get and better complex. Any physician want to get the payment for the service, what we provide, we should do the proper coding. Then only the doctor will get the payment. For example, if I am doing open heart surgery, the code is not in the method of it properly. You can't even assign the proper code, the physician won't get the payment. For example, the surgery is 1 lakh, and you given some other CPD code, which will cost only 50,000. The doctor will lose another 50,000. So what is the code? And this code we are using for different research purpose. data, even statistical data. So they want to know in each country how many people are suffering from diabetes, what is the disease condition mechanism. So all the data also will get with this.
So before going to medical coding, we'll know about RTA, the revenue cycle management, complete healthcare system. In India, healthcare system differs from other countries. Everyone know about it, right? Indian healthcare system is different from other countries because the other countries' healthcare system is more complex and more costly. How many of you know that people from Dubai and USA comes to India to perform certain surgeries? Yes, we know that because that costly. Coming to Indian healthcare, we will go to the hospital if we are suffering from any disease, and we will meet the doctor. We will get the treatment done, and we will pay the bill there. This is all about Indian healthcare. When it comes to US, each and every citizen of US should have signed up for the health insurance. Either it may be a 2 months previous baby or more than 65 years old age people. So once the insurance got done, they will visit the hospital with a prior appointment. Unless until it is an emergency department, they are not allowed to visit the hospital before prior appointment. Once it was done, they will visit the hospital, they will meet the doctor, they will provide their insurance details instead of paying. Then now the complete hospital will come under how the doctor, the physician will get the payment. That is what we are going to discuss now.
So it's the energy of the RT. The RT stands for revenue cycle management. So the patient, so even the patient will everyone have the insurance rate. So we should pay premium to the insurance company. So they will pay the premium to the payer. Payer means insurance company. Patient instead of paying money to the doctors, so they provide the insurance details. They won't pay any money for the physicians. So what providers will do, that is doctors, so they give all the treatment for the patient, but doctor has to get the payment right. So this payment in the form of pay, they will submit to the insurance company. So insurance will check based upon the coverage. And they this is how the in the US healthcare system. So they are the patient, payer, and provider. So payer is insurance company. So if you are not able to quote properly, your physician won't get proper payments. But some is what it
Okay, so the payer means whom we are calling here as a payer? Insurance company, because they are providing payment to the physicians. So here we will be calling as doctors, but in other countries we can call them as physicians. Okay?
So now we go with an example just to understand yourself. Because you are going to educate so many students, right? So if you educate properly, again they will come back here with a success story. That's the reason we have this session now. You can see the sample chart here. So the patient came to hospital. The reason for admission is a knee pain. So the patient came with a knee pain. So once you come to hospital, doctor will take all the history, like how the problem happened, from when the problem is set. So that data is called HPA, history of present pains. Then doctor will take the hours, so they will review the body system, which system it impacts. They will make exam first. And after the review of the system, they have physical examination. Physical, the doctor will catch the areas. They will be able to find out the solutions. Then finally, assessment, which is the problem. The problem something is acute. And further, what treatment here? They give the OTC. They depends upon the patient condition. So, we should read the document thoroughly. We have to understand what is the reason for admission, what a doctor did procedures, and what the final diagnosis is, what the treatment. So this everything we want to convert into simple notes. So now if it is a sample, but we have a lengthy chart. Sometimes some documents we have 8 pages, 20 pages documentation. We have to go through all the 20 pages. Then you have to come on what is the disease condition, what is surgeries, and what is science. So now you can say the answer. What is the scientific international classification disease, right? So the patient is suffering from which disease? In this case, the knee pain osteoarthritis. Because knee pain is a symptom of osteoarthritis. We have to capture the osteoarthritis. This is one of the ELF chat. We had to capture the CPD code. And what the drugs are provided for this challenge? So got a we had to take the code from the HCS. So I'll show you the likes of it. So which we developed from Congress. So we always do the R&D in this software. So where the student will experience the live chat. It is the one interface we developed. You can able to see once you join your medical coding company space, they like in the chat space, department wise. Like they work for inpatient or outpatient or emergency room. So based on what the same chats will be there. The target will be like we had more than 50 chats per day or 100 chats. Depends upon the department. So this is the interface of it goes. So this is one of the emergency department chat here.
You can see
Yeah. So it's all case studies. So in one set of the topic, we can able to see the documentation. In one set, we had to capture the notes. All the topics we integrated the group. You can see the chart. The patient reason for admission is the abdomen. So now we given a live experience how in medical code we'll do the chart coding in live. So what the reason is I have stomach pain in yesterday. That's the reason we came to hospital. Then after taking all the X-ray, the reason history of present illness. Then all the medications we can see, amlodipine, metformin, drugs and taking. Then examination, they come out. They did all the investigation. Then operating. So what the operating they did here? The same like they're starting from appendicitis, right? For appendicitis, what treatment they do? The patient is coming for appendicitis. They remove the cartilage. So remove medical term is called ectomy. So we call it appendectomy. So appendectomy is surgery performed for appendicitis. So we had CPD code which is appendectomy and appendicitis is a disease, right? That will take no ICD. We can able to come out. So we have all the documentation. Disease condition, medication, everything was mentioned. Because medical is best tools for farmers because they know all the drugs. Like these are drugs, they can able to understand what they provide, what disease condition the patient has. So all the condition we need to capture. So now you understand the chart. Right? The reason for admission is abdominal pain. What is the disease condition now to find out? Appendicitis. And treatment provided is appendectomy. So as a medical code, now I read the documentation. Now my role is to convert the document into codes. Right? So now I am converting the codes here. You can see the principal diagnosis. What is the principal diagnosis? This is the software you will be all different types of appendicitis. You can capture them. You can see here the code for appendicitis is A3435.80. So that is the code. So if you give this code, insurance will understand the patient came with appendicitis. What type of appendicitis? So we have a number of specificities will be there. So all this specificity we had to capture. The next patient having hypertension was the documented. We had to capture hypertension because he is taking the drugs. And also is there. So we need to capture that. So whatever the chronic conditions also we have to capture. Because this data will help you for the performance distribution because different processes are there. Like Medicare process and further will be used. And now we done with the treatment. So I see diagnosis. Now what a treatment they provide? With the surgery. So we had to take this as CPD code. So now you can see the CPD code. So we have one column called as CPD column. If you type this code, you can see here only appendectomy is the code 950. Appendectomy will done for indicated, incidental, and laparoscopy appendectomy are open. Because we can do appendectomy by open approach as well as laparoscopy approach. You have to check the documentation what approach they go for. And you have to take the notes. Okay. In case instead of open, you take a laparoscopy code base, dollar value will be very high. The doctor won't get a proper name. Next, we capture all the drugs. You have to check what are drugs, the injections, all things that capture the series. So once you submit, you will go to next process like audit. So in our software, we have here data, which is the correct answers, which codes are missed. You can see here.
These are the capture codes and the fixed codes. Long-term use of insulin, long-term use of drugs. Even we have to capture all the long-term use of drugs. In any patient doing for so many days for chronic condition, that data has to be captured. So all this necessary will be reflected in the software. Show that whether it's 100% coded, 90%. That's how we have our invert open software. The student they can experience that before going live production. I hope now you got an idea how the medical coding works, right? Just offer US certificate charts. We'll get the chart from US. So many companies in India are working on that. Like we have more than 350 coding companies in India work on these projects. So every day they get a projects, they assign to coders. How coders are decided is based on departments. They can work in surgery coders, IT coders. In this, the highest place there is the inpatient coders. Because the inpatient is a tough, complex to make. Because in my experience, I coded charts. Patients stay for 365 days in hospital. When you think about how many charts, how many doctors, the payment was too high. If I not able to code the chart properly, the entire revenue is lost. Because for the one chart, I take around 15 days to code. So very late here. Normally we get like 5 days admission, 3 days admission, but very late here we get like this. Just to code the chart 15 days. So any one mistake, it actually issue a VHS, right? Because I coded the chart, more accurately, and I had put all the numbers. In 365 days, there are nearly 50 doctors involved in this case. So, inpatient, the start if you work for like 1 or 2 years itself, the pay will be 70,000. Because that's such a high demand for inpatient coders, surgery coders. So department wise, the pay will reduce. How the road will be there.
So this is how a day of a medical coder will start. We reach the office. We also worked as a medical coder before starting the business. We will go to the office by 7:45, 8 o'clock is the shift time. We will open our software. We will check how many tasks have been allocated from this department. Because we both work in the same company and we are multi-specialty coders. That means we will be coding ED, ER, and anesthesia, inpatient like that. We will check the charts and we will complete the charts before the shift timing itself. So if we know the proper guidelines as a pharmacy graduates and the teachers, you know well about anatomy, physiology, pathology, procedures and all. So if we know the base and we know the guidelines of the physician, it is very easy to code in a software and complete the charts as soon as possible. So our target is like many of the colleges in India doesn't have a curriculum of medical coding which is pretty much needed to the pharmacy students. So in Tamil Nadu, where we have 5 branches all over the state, many colleges have been taking the medical coding as the value-added courses and the curriculum in their own years. Like for the 3rd year students and the final year students, they have the curriculum of medical coding in the syllabus. We want in India many colleges should take them because one small question: how many pharmacy students is being graduated per year? How many students? I have to remember. Sorry? 100. So all are getting the dream job what exactly they wanted for? Yes? Very good. So if yes, that is pretty much great. If no, there are so many fields to choose. Like healthcare. Nowadays, healthcare, the total description has been changed. Healthcare is nothing but patient, hospital, clients. A client is nothing but a patient medical record, getting the payment to the physician. This is all about the healthcare system nowadays changed. So any of you visited any foreign countries or know about the healthcare there? How it will work?
So coming for the growth here, because it's like around like 4.5 trillion business sector in the industry. As it's a more volume, it's getting outsourced to India. The main reason for outsourcing for India is skill. We have a great skill to process any complex charts. Okay. So along with the time zone, time is one of the advantage and the cost effective also. The same chart, if you want to work in US, per chart they have to pay $1, but they're not paying $1 per chart because we used to go like 50 to 100 charts per day, which is a lesser pay. That's the reason we get like more dollars also. Even we run a coding company. We do work with the physician tools and aggregator projects. So what are we are giving back the same people we are hiring in our own companies. That's all the capability they get. And coming for Indian charts, why we are not coding for Indian charts? Because most of the people are thinking that we are coding for Indian charts. We are not coding Indian charts, we are coding only US charts. But we have a new scheme introduced by PM Modi, right? So which is Ayushman Bharat Digital Mission. In a time of 5 to 10 years, even everyone in India also will have health insurance. If health insurance everyone has, then we will put our charges. Because the insurance company want to pay the money, right? They can't just simply pay by reading the documentation. They want a uniform system. So in India also shortly we will introduce that. Then we have more companies will be there for our field. This is a debate. Already discussed the chart volumes, we have more volume also stream here.
So this is the second part of medical coders. They will be joining as a junior medical coders and then the senior medical coders, and they can choose any specialties like inpatient surgery, anesthesia, like that. And they will be promoted as a quality analyst or an auditors, and then they will be promoted as a trainers and managers. This is how the growth will be, but purely depends not on the seniority but on the performance and talent and complete knowledge in medical coding. If we have proper knowledge and talent, within 6 months also you can go as a manager. Because the maintenance skills and the people management skills, if we have and if you are doing a quality chart, you will get a promotion as soon as possible. This is completely based on the performance.
It's all the salary varies. So starting depends on the certification, non-certification. So it will start from 80,000 to 25,000. It's a company, some companies, MNC companies, base salary. So year on year, we are 30 to 40% high, and depends on the department. Education is a high scope, but more complex is it. So we need to achieve the commerce because commerce students are best suited for inpatient charts. I already said because of the drugs. As complex drugs coding is not easy for other graduates, it's easy for the pharmacy graduates.
With AI replace medical coders, this is the most happening question right now. No, AI is not going to replace the medical coders, but it will foster the work. Instead of your reading each and every document for 20 minutes, it will do 100 records reading within 10 minutes. But the human work is to do the accuracy in the coding. Because if you are not giving the proper appropriate code, the physician patient will affect them. If, for example, if the physician has done CT, instead of CT, if we give an X-ray code, is it going to affect the payment of physician or not? Right? So the AI can read the document faster. He can't analyze properly what the doctor wanted to say to the insurance company. That is the reason why we can foster our work with AI, but it is not going to replace the humans.
So what they should have is to enter into medical coding is, so they should have the basic knowledge in anatomy because we should read the documentation. Right? Then the person should have all the knowledge in medical technology, then usage of the books. We have 3 books, right? So they should get it in-depth knowledge in each of the books. Then they should know the coding techniques because every doctor is having separate techniques. When you work for US clients and a group of physicians, we have one set of techniques. We had to follow those guidelines. So after that, we had to do the prediction. And you should know how to use the different softwares because it's more on like technical jobs. You're not going to see when you come back here. So you should have a hands-on experience on the softwares. So using that only, we can able to code that. why it best suits for pharmacy graduates is already pharmacy graduates are doing anatomy, physiology, pharmacology, pathology, medical chemistry. That is already there 80% ready to code each. There is a most of the companies will hire pharmacy graduates. But for students, there is no much awareness on this because after graduation, I went to different streams. Okay, and even I was like 1 or 2 years. Then I realized that yes, we need to stabilize any one domain. Stabilize, we should be in a top position. That's how we keep on work. We are in this position now because of sticking to one domain. The same thing get taught in the college. Definitely, we will make immediately after college, we will get a job opportunity. The only intention we come and inform all the college and if any students are interested, we can make them to go into coding based upon their resources. So how the target will be there is depends upon department. In case if any coder is working inpatient coding is then daily target is 15 to 20 chats. That means they do 20 patient case studies. By end of shift time, they have completed 20 cases, then they get paid. In case any coder is doing extra charts, for extra charts, extra incentives will be there. Okay, because we have only 5 days of working, so that is when they want to work, they to make it to work, they can do. And extra charges also they have extra pay. So that's how we department very easily get code, like various department. Various departments, say what number of views, and they can capture very fast. In those departments, the chart number will be high. So coming for our academic topics, main intention is like to give more awareness of this domain because India requires skilled manpower in this domain. We also run the coding company. Always we find issue is we are hiring the people that not up to the mark in terms of production. If there is a production, so we had to pay the money for the doctors. I'm getting a project from US, but don't the payment. Is not making the quality we have to give. This is a company where we used to do all the production, which is thoughtful, healthcare, project limited. So it takes to make ready the performance graphics in different teams of operations. That's all from our end. Thank you very much. I hope it's an excellent time.
Thank you so much for giving this opportunity for the Offers Mental Coding Academy. We came long from Congress here yesterday to attend this event because we both are from Andhra Pradesh and it is a border of Andhra. We really very happy to deliver this lecture today to all of you guys. Thank you so much once again for providing this opportunity.
On behalf of Health Sciences, we extend our hearty thanks to all the team members, Mr. Madam Bhanwati and Madam Viradari and Murali Sarji. Thank you so very much to one and all. We extend our thanks and recognition for the pay taken with the traveling long distance. Yeah, I hope also we had interaction with students for whom it will be more useful, definitely. So without, so you also will be feeling hungry and all the delegates, teachers, our expert person, all are so now it's time to felicitate and further interaction. So I like to request our principal madam and Sarati and research persons are two quite great. Sagar sir and this other sir on the back. Request Prasad sir also to join our felicitation and Amana sir please come on and sir. Request Prasad sir please come on the back and felicitate all the three guests. Second and please. We are all the we want to minister.
Thank you all the participants and thank you all the institutes in cooperating this program. Now it is time for lunch. Every day is getting late. Please join for lunch. Thank you. Thank you so much.
The time.